So basically it could be a prediction or a scope, but let it be a guideline for you to prepare for your exam. And I know that by the end of this lesson, you must be able to get a distinction. Those who have the distinction material book, I know that the distinction is already in your hands. All right, let's start without wasting time. The first question. The bond that forms between the amino acids during protein synthesis. How do you call that bond? So we call that bond peptide bond. Peptide bond. An organelle. You know, I like to start with the terminologies because this one, it warms you up so that you're able to prepare for the exam, or the terminologies of the terminologies of the paper. All right, the organelles that contain DNA, which is used to trace the female ancestry. Remember, we have 3 organelles which contain DNA: nuclear DNA, the nucleus, which you call the nuclear DNA, then you have the choroplastic DNA, so it's found in the chloroplast, then you have the mitochondrial DNA, which is found in the mitochondrion. But it's only the mitochondrion which is used to trace the female ancestor. Therefore, the answer is going to be mitochondrion. Right. The saying, undifferentiated cells and differentiated cells in animal that have the potential to become any type of tissue. It means that they can develop into other body cells. So we call them stem cells. A change in the sequence of nitrogenous bases resulting in the formation of a different protein. So such thing, since it is change of the bases, so it's gonna be point. You can call it point, yeah, because the bases are specific. You can call it point, or we can call it gene. Yes, gene, but because they're saying that change in sudden change in science, we call it a mutation. So in actual sense, maybe the answer could be mutation, or gene mutation, or point mutation. So yeah, natural shape of DNA, always they ask you what is the natural shape of DNA. No any other shape. We call it double helix. The permanent disappearance of species, from the earth. So how do you call it? We call it extinction. Yes, the type of variation where there is a range of intermediate types. For example, if you have look at the size, you have big, small, medium, extra big, or extra XXL. Call it XXL. Then we have the small. So a range of phenotypes, but it's one character. So we call it a continuous variation. So when you say that that type, that's the variation, and then it's saying range, so it is continuous. You have different range of this character. Very dark-skinned, light-skinned, very light-skinned. You see, it's a range of character, but what gene is it? Which character is it? That is skin color. Yes, so it becomes continuous variation. An allele that is expressed in the phenotype in a heterozygous state, meaning that if you look at the heterozygous state, for example, capital T, small t, this allele, it means that if T for tall and small t is for short, so if the individual is like this, capital T, small t, this individual is gonna be tall. So how do you call the allele for tallness? So we call it, how do you call it? We call it dominant allele. So it's dominant allele. Yes, we call it dominant allele. The position of an allele on a chromosome. How do you call the position of an allele? Or a position of a gene on a chromosome. You can change it and it's the same thing. So we call it locus. If there are many, we call them loci. But since one is called locus. Let's go to other types, and then we see, I know that you have not yet feel like you are ready for these terminologies. So let's have another example. The type of inheritance in which produce intermediate phenotypes, you see. So you produce intermediate phenotypes. So how do you call that kind of dominance? So we call it incomplete dominance. Why do we call it incomplete dominance? For example, you have black crossed with white, and then you produce an intermediate color which is gray. So how do we call this type of dominance? How do we call this type of inheritance? So we call it incomplete dominance, because neither of the allele is completely dominant over the other. If you look at the previous one, we see that tall crossed with short, and then you get what we call tall, meaning that tall is completely dominant over short. So the point where adjacent chromatids overlap during crossing over, so we call it chiasma. If one is called chiasma, if they are many, we call them chiasmata. The process where information is copied from DNA to messenger RNA, how do you call that? We call it transcription. Formation of the messenger RNA. From DNA, we call it transcription. The sugar found in DNA molecule, how do you call that sugar? Deoxyribose. From the name DNA, which means deoxyribonucleic acid, meaning that it has a sugar called deoxyribose sugar. The bond that occur between nitrogenous bases in the DNA molecule, how do you call those bonds between the nitrogenous bases? We call them hydrogen bonds. So to be more specific, call it weak hydrogen bonds. All right. Manipulation means that you are using the man. That's why you call it manipulation of organisms gene. When you manipulate, actually you are an engineer. Yes. So it means that if you are an engineer, yes, then it means that what are you engineering? You are engineering genes. Therefore, manipulation, engineering genes, how do you call that? To bring the desired characteristics. So the answer is already there. So we're going to call it, since you are manipulating the genes, therefore it's going to be genetic engineering. The representation showing the arrangement of a diploid set of chromosomes. How do you call representation showing the arrangement of diploid set of chromosomes? So if you look at them, meaning that there are sets from chromosome pair number 1 to the chromosome pair number last. So how do you call that? Since you are describing the nucleus, so the nucleus in science is called karyotype. But because you are looking at the type, how are they arranged? So it's going to become, which type of nucleus is that? Hence the name karyotype. So we call it karyotype. All right, let's go to match column example, and then we see if you are ready for this paper. Source of variation in the species. Random arrangement, random mating. Remember, random mating, you don't have to be specific that I want the one which looks like this, I want the one who looks like this. Whoever comes, you go with that. Random mating. Random fertilization. How many sperms are being produced in a single ejaculation? Over 400 to 500 million sperms are being released in a single ejaculation, but only one and you don't know what is that one. So since there are a lot and we don't know which one is gonna do so, it's randomly done and each sperm is different from one another. Therefore, we're gonna call it random fertilization. And this is also a key for bringing variation. That's why your brother might look different from you, because of random fertilization. Therefore, both of them, they contribute to variation. So the answer is both A and B. Organisms possess, an organism possesses two factors which separate so that each gamete contains only one of these factors. How do you call that? Law of inheritance, law of dominance? Not really. Principle of independent assortment. Organisms possess two factors which separate so that each gamete contains only one. Law of principle of independent assortment. Not really. So the answer here is gonna be none. Reproductive isolation mechanism in species. Breeding at the same, this makes it to be wrong. Same season? No. They can't be separated if they are breeding at the same season. Infertile offspring. Infertile offspring, meaning that you produce like a horse and a donkey, then you produce a male which is infertile. So this looks to be B only, and you get your 2 marks, 2 marks, and 2 marks. Then we go to the next question and we see what is it talking about. So if you look at this section, it's still giving you a dose to see that, yeah, the dose is enough so that you don't have issues with the terminologies. A section of DNA, the DNA molecule that code for a specific characteristic, we call it a gene, is a section of DNA that control a specific characteristic or a particular characteristic or a particular trait. Sometimes we call it trait. Yes. Pattern of black bears resulting from DNA analysis. So how do you call that? They're not looking at the process, they're looking at those patterns you produce. So we call it DNA profile. The process is DNA profiling, but the results is the profile. But whose profile? For DNA. Evolution characterized by long period of change and rapid period of no change. How do we call that? We call it punctuated, yes, equilibrium. Punctuated equilibrium. Yes, punctuated equilibrium. Okay, they're saying that the production of genetically identical copies of organisms, how do you call that? Like you are exactly the same. So you, in science, you call it you are clone. You are a clone. You are a clone. So the process is gonna be cloning. Yes, undifferentiated cells in animal that have a potential to become any type of cell. So we say that we call them stem cells. So you can't remember this, then it means that you have a problem with cerebrum. A genetic disorder that results in a person cannot distinguish between colors. How do you call that genetic disorder? We call it color blindness. Call it color blindness. The part of plant where male gametes are produced. Remember, we call them anthers. Anthers. Yes, I don't want to go to the distinction material book to show each and everything because it will be taking a lot of time, but I think you know what I'm talking about. The structure that joins two chromatids together. How do you call the structure that joins two chromatids together? Central. That structure joins the two chromatids together, the central body, centromere. Okay, then we are saying that The type of dominancy, neither in which neither of the two allele is dominant over each other, resulting in offspring with an intermediate. Who can tell me the answer? Who can tell me the answer? Who can tell me the answer? Intermediate. Here, the first one, you have the complete dominance, you have the codominance. I think you didn't get it right. The answer is none, because we talked about it before. So that's why I put these questions to check you up, see that you are able to remember. All right, so none of this, because this one brings a mixture of two, and then this one brings the one which is dominant becomes expressed in the phenotype. The point where two chromatids overlap during crossing over, what's the answer? You forgot. The answer is chiasma, and I said if there are many, we call them chiasmata. The scientists who produced the law of segregation, what's the answer? None, because this one, Darwinism, Lamarckism, they are in evolution. All right, so the one who did that is Georg Mendel. All right, the position of a gene, on a chromosome, what did we say? You forgot. Is locus. The type of evolution characterized by long period of change and followed by a short period of rapid change. What did we say? Punctuated equilibrium. If you forgot the natural shape of DNA, what did we say? Double helix. If you guys can't remember what we said, we just said double helix. The type of bond found between two amino acids, what did we say? Peptide bond. The type of vision which is shared by primates that allow deep perception, how do you call that? Is the binocular vision. Yes, or stereoscopic. Right, the type of dominance in which results in the intermediate of phenotype, what did we say? Incomplete dominancy. Yeah, you guys, if you are not able to remember what you have said, then means that you need to catch up. I have to go to the catch-ups, and we talk about the catch-ups. Then the fluid of the nucleus, where the free-floating nucleotides are found, how do you call that? Nucleoplasm. Entangled mass of chromosomes located within the nucleus, how do you call that? We call them chromatin network. The division of the cytoplasm after nuclear division. How do you call the division of the cytoplasm after nuclear division? When you say division of the cytoplasm, division or splitting, or how do you call that? Does it require energy? All right, yes. So since it's a cytoplasm, so I will call it cytokinesis. The name for X and Y chromosome in the human, we call it gonozome. So guys, that is our first section of the terminologies, and I think guys, you are now well to go, and then kill the paper, because the paper should not kill you. Let's go to the first section of that. But before I go there, don't forget this. Yeah, so your paper 2 will be will be DNA, meiosis, genetics, evolution. And if you look at this, check the marks you're supposed to go for. Yes. All right. So we continue to the next question. So if you look at this question, they are saying that process shown in the diagram above. Which process is shown in this diagram? So if you look at this process, we shall see that this process is, let me do like this. Which process is that? If you look at that diagram, we see that this is DNA replication, because you have a new strand here, you have a strand here, old strand, and then you have an old strand, and then you have new strands being formed. And because of this weak hydrogen bonds, it means that each molecule is DNA. Therefore, I will say that this process is called DNA replication. Chemical bond between labeled 1. Chemical bond labeled 1. If we clear this up, let's find out which chemical bond is labeled 1. So these are weak hydrogen bonds. Weak hydrogen bonds. Yes. Then we got the next question saying that collective name 1 for 2, 3, and 4. 2, 3, 4. So the answer here is gonna be the collective name. They are looking for the collective name. So the collective name is going to be the nucleotide. Full name for nitrogenous base 5. What is 5? 5 is here. Here you have adenine. Automatically it's going to be thymine. So 5 becomes thymine. Okay. Thymine. All right. During interphase, during the phase of cell cycle, does it take place? When does it take place? So basically, this takes place during interphase. Where in the cell does this process take place? It takes place in the nucleus. So guys, yeah, that is the questions which let's go to the second question. But before we go to the second question, you need to know how to label. Basically, this is DNA, and this process is transcription. So you have messenger RNA being formed. So R is messenger RNA. 3 is the amino acid being brought. Yeah. It is amino acid, and then this is the messenger RNA aligned itself on the ribosome. Remember, sometimes it's supposed to have 2 bases, the minor subunit and the major subunit. And then you have the anticodons, then you have the codons on the messenger, and then here is the nucleus, and then here all this place is the cytoplasm. So once you know that, now you are able to go and try to answer the questions they have asked you. Okay, so we go back and ask ourselves, identify process Z. Identify process labeled Z. So if we check here now, we are saying that what is Z? Which process is Z? So we say that Z, the process of Z is transcription. Then molecule R, where is molecule R? Is this, which is a messenger RNA. And then we're saying that organelle Q, this organelle, so it's gonna be ribosome. Give the collective name of O. Give the collective name of O. We say that these are anticodon. It's anticodon. Determine the sequence of nitrogenous bases at area S. Area S. They're saying determine the determine the sequence of nitrogenous bases at area S. So when you go back to area S, we shall see that T goes with A, C goes with G, and then A goes with T. So it's gonna be AGT. Why? Because it is DNA versus DNA. It's not messenger. That's why there is no U. Then they're saying that which strand was used as a template. Which strand was used as a template? So if you look at, you have strand 1 and strand 2. So we see that what is formed here, that's strand 1, and then this is strand 2, of which this strand, they say that if you look at it here, we need to check out here. What does this A, because this is messenger, if this is messenger, what does this one correspond with? A goes with the T, and then U goes with A, and then C goes with the G. So I'm going to look for the strand which has this, because this is a mirror for this. Therefore, this became a template. TAG. So it is strand 1. Shows that strand 1 is the one which is used as a template for that case. Then they are saying that which amino acid 3, 4, 5 will be brought by P. Will be brought by P. We go back and find out what is P. So we see a P. I don't know there. Now I need to go back, and then again I only have this and this. I don't have it here. Cool. I will have to go back again, because now this and this is the same, ne? So I have to go back here and I find out, because this is strand this is strand 1, but still I don't have it here. So it means that to get this, because this and this is the same, to get this I have to know what is this, but to get this I have to know what is this. Therefore I'm gonna say TCA, that is for this. Now we need to find out what is this. So it's gonna be AGT. So this is this. Okay, now once I know this, now I need to find out what is this. So it's gonna be A goes with U, because it is tRNA, CA. You see? So now, since now I know the messenger RNA here, I need to know the transfer RNA, So the UCA will go with AGU. So I have to look for AGU. AGU is here. So amino acid number 4 is the one which is gonna come. Amino acid number 4. They are saying that name the type of sugar that forms part of the structure molecule R. Where is structure molecule R, part of sugar? So we said it ribose sugar. Yes. Then we go to the next question of DNA again, and then we see the diagram represents the stage for protein synthesis. Okay, so if you look at this, since you see that this is a ribosome and this is a messenger, then automatically this is a translation. So it is translation. So now, since that is translation, so now it means that identify the organelle 3. We say that organelle 3 is a ribosome. Then describe the role of molecule 1. Molecule 1, Describe the role of molecule 1 during the stage of protein synthesis. So basically, molecule 1 brings the required, you see, it's bringing the required amino acid to the ribosome. So it brings the required amino acid to the ribosome, or brings a specific amino acid to the ribosome when its anticodon matches the codon of the messenger. So it brings the required amino acid to the ribosome when its anticodon, or when tRNA anticodon matches the codon of the messenger. So that's how you're supposed to explain that. Give the sequence of nitrogenous bases at number 2. So at number 2. So number 2 is going to be CCU, because this is messenger, it is transferring A. Since it is RNA, so it's, A goes with U. Then they're saying that on the DNA molecule that code for histamine, give the sequence of nitrogenous base on DNA. Bases on DNA that code for histamine. So if you look at this, actually it's supposed to be replaced with the T. Okay, sharp. Let's find out. Since this is tRNA, so I'm looking for DNA. So G goes with the C, this goes with the A, and this goes with the U. That is messenger RNA. This is transfer RNA, this is messenger RNA. Now I need to convert messenger RNA to DNA. So it's going to be GTA. GTA. So that's why I said this is replaced with the T. So it means that it's gonna be GTA. So I have to convert it into 2 times, because it is, I'm changing from tRNA to messenger RNA to DNA. During the synthesis of the same protein, there was a change in the amino acid sequence, because alanine was replaced with tryptophan. Alanine was replaced with tryptophan. Explain the possible reason for this. So if this happens, so we call this a mutation. So it means that it was due to a mutation. So you mentioning us that it's a mutation, is a change, you might get a mark. So what is other 2 marks? So they say that alanine, this alanine, of which alanine is GGG, was replaced with tryptophan, which is GAA. Yes, that means that during the DNA sequence, changed from GG to GAA. That's the meaning. You understand? So basically it means that the G's were replaced with A's. You see? Let me check properly. Alanine was replaced with tryptophan, meaning that there was alanine was replaced with tryptophan. So that's the meaning that the GG was replaced with the AA, making it to be a different amino acid. So basically you have to tell us what really happened. If you see that alanine is coded by GG and the tryptophan is coded by GAA, then it means that the G and G was replaced with A and A, and then because now on DNA molecule therefore resulted in different amino acid, and this process of changes is called mutation. That's why your first mark at least is going to be going to the mutation, and then you tell us what exactly happened, that GG was replaced with AA, which resulted in the formation of a different amino acid. So there is no way you're gonna skip a question concerning about mutation. So just know that a question for mutation is gonna come. So please, when it comes, make sure that you answer that question very well. Then we go to the next question. What is X? Is a amino acid. What is Y? Y is 3 bases. So I will call these ones anticodons. And then Z, if you look at Z, Z is going to be the messenger RNA. So which process is this? Basically, this is a translation process. So they're saying that identify molecule X. So if you look at it, molecule X, we said molecule X is amino acid. Z, we said Z is a messenger RNA. Give the nitrogenous base sequence of DNA triplet complementary to the middle codon on Z. So if you look at this, it's messenger. They say the middle, the triplet which code for the middle is so it's gonna be on DNA. So it's gonna be A goes with the T, U goes with the A, and then G goes with C. So it's gonna be TAC, which means that TAC. Nitrogenous base sequence of Y. Where is Y? So in this case, since I have this CAU, so I'm gonna convert into transferrin A, which is gonna be G, GU, and then A. So that is going to be the answer for Y. Name the process and describe the process. Name and describe the process shown in the diagram above. So in this case, we want you to explain translation. So if you go our distinction material book, it is clearly stated there, or according to the messenger RNA codon, the transferrin A with the complementary anticodon will bring the required amino acid to the ribosome. The amino acids are joined together by peptide bond to form a required protein. So that's what you support. You can't go to the paper without knowing the transcription, the translation, the DNA replication. You can't do that. If you do that, just know that you are messing yourself up. All right, so we got the next question. So the table below shows the codon, shows the codons that code for some amino acids. A mutation caused by DNA base triplet to change from CTG to CTA. Describe the effect of this mutation. So if there is CTG, which code for triplet on DNA, what do we have here? We have messenger. So if it is CTG, then it's going to be CTA. I need to first convert this to because this is DNA, and what I have here is messenger, so I have to convert into messenger RNA. C goes with G, and then T goes with A, and then C, and then this one I also have to convert it, so it's gonna be GAU. So what does this code for? GAC is aspartic acid. So this is aspartic acid. And what is GAU? GAU is also aspartic acid. Even this one is aspartic acid. So it means that there is no change in the protein. Therefore, I have to tell that the codon the codon GAC, yes, changed on the messenger RNA, changed to GAU. You understand? Both they code for the same amino acid, aspartic acid. Therefore, there was no change in the protein, or there was no effect on the protein which is formed. Yeah, so in that case, you have to first identify the sequence which is being given here. Is it DNA or messenger RNA or transfer RNA? And then what is given here, is it DNA, messenger RNA, transfer RNA? If it is the same, you keep the codes. If it's different, then you have to convert so that they are the same. So in this case, what is being given is DNA, and what was given here is messenger. Therefore, I had to convert from DNA to messenger so that I'm able to match them, and then I read them. If I find out that the amino acids are the same, coded by these codes, therefore there is no change in the protein. But if I find out that this code is for aspartic acid and then this code is for leucine, for example, then it means that there will be a difference in the amino acid, therefore there will be a change in the protein. Then we go to the next question. Tabulate difference between DNA and RNA nucleotide. Here they are talking about nucleotide, but if they ask you to tabulate, make sure that you draw a table. The moment you draw a table, that's 1 mark. And if you draw a table, don't just draw a table which is just empty, like T like this, 0. So make sure that DNA, it must be complete, nucleotide, RNA, nucleotide, then we'll give you a mark there, that's 1 mark. Then point here, DNA has ribose sugar, DNA has deoxyribose sugar, RNA has ribose sugar, you get a mark. DNA, you tell me, has thymine base, RNA has, what does it have? Has uracil base. Then you get another mark. You get 1 mark here, you get 1 mark here, you get 1 mark here. That's all. Make sure that whenever they ask you to tabulate, don't just draw a table, which some of you just draw because you want to finish and leave, which is wrong. You are messing up your life. Yes, someone was saying that the warmer the blanket, the colder the future. I liked that statement when I was busy online. I like that statement. The warmer the blanket, the colder the future. Yes, that's it. So if you like the blanket too much, just know that the future is going to be colder. And if you leave the blanket so it will become cold, then it means that the future is going to be warmer. So that is it. The next question is about the DNA profiling. Name the technique which is being used. Is DNA profiling. Then they're asking you which suspect 1, 2 was at crime scene. If you look at here, says sample, suspect 1, suspect 2. This is the sample from the victim, and then this is a sample from the crime scene. So it means that we have to match. So if you look at this, matches with this matches with this, all of this match with this. So automatically suspect 1 was at the crime scene. Give one visible reason for that. If you look at it, the DNA profile, you have to, don't just call them DNA DNA profile or DNA bands. Please don't change the words. Bands of suspect 1, yes, is identical to the DNA profile in the DNA profile of the crime scene. You see, DNA profile of suspect 1, yes, is identical or is the same as that of the crime scene. So that is the reason why we gave that to be the answer. Then they're saying that give 2 possible reasons why the suspect identified question might not be convicted if the evidence is used. First of all, yes, we know that DNA is unique, but it might be manipulated. What if someone went there and then dropped it? There could be human error. The suspect can say that no, that is human error. It could be contaminated with something else. You understand? Yes, maybe they used a too small amount of what did they use? Blood? What? Small sample. They used a small sample from the crime scene. Can say that no, that's not mine. You understand? Yes. So that's some of the reasons that might not be convicted. Then they are saying that explain why the technique mentioned above is used in the paternity test. Explain how. They're asking you how this technique is being used in the paternity test. So basically, if someone is claiming that this is my child, you have to first identify the DNA profile of the child and the DNA profile of the mother. You understand? The DNA profile of the child and DNA profile of the mother. So you remove all the DNA, the bands which match with the child and the mother. The remaining bands of the child must match with the father, the possible father. And if they match, therefore that will be the father. But if they don't match, then that suspected father won't be the father, because they don't match. So DNA profile of the mother and the child, number one. Number two, match all the DNA profile of the child with the mother. Once that's done, now remove the remaining profile, must match with that of the must match with that of the father. If they match with that suspected father, then automatically that is the father. If it does not match, then it means that that is not the father. So now from here we're gonna go to what we call our catch-ups, and then we see some of the questions in the catch-ups. So if we go to our catch-up, we see the question, describe the structure of DNA. So in this case, you have to describe how DNA is structured. Remember, we say that DNA is made up of nucleotide. Yes, made up of what? Nucleotide. Nucleotide is made up of phosphate, deoxyribose sugar, and nitrogenous bases. Then you tell us adenine, thymine, guanine, cytosine, whereby adenine pairs with thymine and guanine pairs with cytosine. And then you tell us, you just basically describe the structure of DNA in that regard. It has two strands joined together by weak hydrogen bonds. So you can't go to the paper without knowing how to do the proper description for this structure. So if you go back here at the beginning of DNA, you just look at the structure of DNA, and then you are able to describe that structure based on what you already see. You just have to know that that's the structure, and then you have to describe the structure. Sorry. So now we got the next question. They are saying, what's the next question? What are the monomers? Of DNA? We said they are nucleotides. Who discovered the structure of DNA? That's James Watson, and then Francis Crick. If you look at the people who are involved, then automatically you have to also include Maurice Wilkin, and then Rosalind Franklin. So those are the four people who are involved. Who discovered the structure of DNA using X-ray diffraction picture? That is Maurice Wilkin and Rosalind Franklin. Then they are saying that in which year was that? It was 19. Basically, when was the Nobel Prize being given? So that was being discovered in 1962. So other than nuclear DNA, name other types of DNA. We have what you call the mitochondrial DNA, the one which is found in the mitochondria, and then we have what you call the chloroplastic DNA. What is DNA? Replication is the process whereby DNA makes exact copy of itself. When does it occur? It occurs during interphase. Where does it occur? It occurs in the nucleus. Why does it occur? To increase the chromosome number. So basically here you look at the distinction material book, and then you see that all these things we are talking about, they're there. How does it occur? Here you describe the structure of you describe the process of DNA or replication. So that's what you're supposed to describe. So for example, in this case, what you have been talking about is here. What is DNA replication? When does it occur? Where does it occur? Why does it occur? You see, double the chromosome number, And then how does it occur? You explain DNA replication. Double helix DNA unwinds, weak hydrogen bond break to form two separate strands. Each is acting as a template using free floating DNA nucleotides. Remember, in transcription we used free floating RNA nucleotide. Here we use free floating DNA nucleotide from the nucleoplasm. A complementary strand is formed whereby A pairs with T and then G pairs with C. Two new DNA molecules are formed. This DNA, they are genetically, they might not be, they swap the strands, but if you look at them genetically, they're identical. So you're saying that is identical and someone else saying that genetically identical, those are two different stories. With one original strand and one new strand, this process is controlled by the enzyme. So you get the marks for that question which asks you to describe how does it occur. What are some of the functions of DNA profiling? This one is a simple thing. I don't usually go through them, but we can use it for paternity test, identification of individuals at the crime, the crime or crime identification. You can talk about diagnosis of inherited disorders. You can talk about personal identification or relatives, maybe, or for example, when someone is dead or burnt, so we are able to identify that individual. So that is DNA profiling. What are disadvantages? Sometimes we call them debates about, or what are the debates about DNA profiling? Very expensive. Number two, sometimes a person they cannot, it might not be 100% correct due to the human errors. You understand? Human errors. Not all hospitals have the necessary equipments. Can also lead to the fault results. You see? Yeah, sometimes you can also frame the individual at the scene. Someone did it and then dropped something which belongs to you, you see. So all this can be a reason. What is the function of messenger RNA? It carries coded information from DNA to cytoplasm so that a protein can be formed. So it basically carries coded information from DNA, that is from the nucleus to the cytoplasm, so that a protein can be formed. Why is the messenger RNA has a specific sequence? Or basically, what's the function of the messenger RNA? Why is the messenger RNA has a specific sequence? This one is basically to determine the specific sequence of these amino acids, hence a specific protein. So the specific sequence of messenger will determine the specific sequence of transfer with its amino acids, and then it will determine the specific protein. Then we go, what is protein synthesis? We have talked about all this, but we are just doing a recap or a catch. Then what is DNA replication? What is protein synthesis? Is the process whereby DNA makes exact copy of, is the process whereby the cell makes proteins. The process where proteins are being made in the cell, process where the cell makes proteins. So this process is divided into two, that is transcription and the translation. Okay, and then transcription is the formation of messenger and translation is the formation of polypeptide chain. Which organelle does transcription take place? Organelle occurs in the nucleus. The organelle is nucleus. That is where transcription takes place. On which organelle does translation takes place? It occurs on the ribosome. ribosome. Describe the process of transcription. Describe the process of transcription. Here we talk about double helix DNA unwinds, weak hydrogen bonds break to form two separate strands, one acting as a template for formation of messenger RNA using free floating RNA nucleotide from the nucleoplasm. A complementary strand is formed whereby adenine goes with the pairs with uracil and cytosine with guanine. Now messenger RNA has a coded message for protein synthesis, which moves out of the nucleus through the nuclear pore to the ribosome. You see, that is transcription. That is transcription. So you have to know the process of transcription. And then what is translation? According to the messenger RNA codon, the transfer RNA with the complementary anticodon bring the required amino acid to the ribosome. The amino acids are joined together by peptide bond to form a required protein. So equivalent is a key point. So you guys who are using distinction material book, I think now you know what you're supposed to do. Please use it effectively so that you're able to succeed effectively. What is the difference between DNA nucleotide and RNA nucleotide? When you talk about DNA nucleotide, nucleotide of DNA, we shall see that this nucleotide, they are not asking about the strand. It has thymine for DNA, for RNA it has uracil. It has deoxyribose for DNA, for RNA has ribose. Yes. So that is it. What is the difference between the DNA replication and transcription? Here you look at what happens in DNA replication but does not happen in transcription. Here you form two new DNA formed. Here you form a protein at the end. Here we see that we use free floating DNA nucleotide. Here we use free floating RNA nucleotide. This one occurs in the nucleus only. This one occurs. Oh, they say transcription, not protein synthesis. So it means that here occurs in the nucleus. Also, this one occurs in the nucleus. Oh, yeah. So at the end of the day, here you have formed two new DNA. Here you form messenger RNA. Here you use adenine goes with thymine. Here adenine goes with uracil. So basically there are a lot of differences, even similarities. Here we see that we have DNA double helix DNA unwind. Even here double helix DNA unwind. Weak hydrogen bonds break. Here even weak hydrogen bonds break. This reaction occurs, or this process occurs in the nucleus. This process occurs in the nucleus. So you see that there are similarities which are happening between them. So from there we have to go to what we call meiosis. But before we go to the catch up of meiosis, we have to go back and see where we stopped with the meiosis. In this case, we're going to start with what we call the karyotype. So this karyotype, if you look at it, we see that the karyotype, does it have any abnormalities? It doesn't have any abnormalities. Oh, it has here two of them, but the others are single. Okay, name the type of mutation that occurred during the production of this gamete. So it shows that the type of mutation is chromosomal mutation. Chromosomal mutation. What type of gamete is represented by the karyotype? Because of X and Y on 23, so it means that it's gonna be a male. In this gamete, give the number of orosomes. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22. So there are 22. There's no one which is mutation where there are 2 of them. So there are 22. Gonosomes, it's 1, 2. So it's there 2. They're saying that the gamete, remember gonosomes are the sex chromosome. The gamete fuses with the normal gamete. Gamete fuses with the normal gamete. Using X and Y representation. Write the sex chromosome of the offspring. Sex chromosome of the offspring. Now the question here, the challenge here is that they did not specify which normal gamete. Oh, normal. It means that if it is fusion, then it means that it fertilized the normal gamete, which is the female. Yeah, so there's no challenge. So since this is X and Y, so it's going to be X and Y, yes, then plus a normal gamete, which is going to be, remember the normal individual is going to be like this for the female, only one comes. So it means that this individual at the last is going to be XX then Y, XX then Y. So that will be the phenotype of the genotype of that individual. Then we go to question number 4. The diagram below represents a cell in the early stage of meiosis. So here we see that this is a cell nuclear membrane breaking, then this is cell membrane, this is nucleoplasm. Then they're asking the phase of meiosis represented. The phase of meiosis represented. So if you look at this, you see that crossing over is going to take place soon. So it means that this is prophase 1. Name the chromatid shown. Number of chromatid. Number of chromatid is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. So basically number of chromatids are 12 because each chromosome has 2 chromatids. It has this and has this. So number of homologous chromosome pair, if you look at them, this 1, this 2, this 3. So the answer is going to be 3 chromosome pair. Then they are saying that identify structure. R, we say the nuclear membrane. S, we say that is cell membrane. And then T, we said is the nucleoplasm, the fluid of the nucleus. Name 2 organelles in the animal where meiosis takes place. 2 organs in the animal where meiosis takes place. It occurs, they say the animal, in individuals. So it takes in the testes, yes, and also it takes place at the ovaries. Yes, those are the 2 places where these individuals, where this can take place. Then from there we go back to our catch-up, and then we see what is on that side of our catch-up. There are a lot of questions you can do, yes, but crossing over, you have to know the process of crossing over, you see, yes. So let's go back to our catch-up and then we see. So this is meiosis. I hope you guys, I said that this mutation will be coming, so make sure that you prepare that mutation properly. So when you go back to our catch-up of meiosis, give 2 importance of meiosis. Meiosis, first of all, the importance of meiosis is very good for variation. It's very good for variation. Number 2, production of haploid gametes. Number 3, those things are here in the distinction material book. I don't want to go there, you understand. The doubling effect, solving the problem of the problem of what? The doubling effect solves the problem of the halving effect of meiosis solve the problem of doubling effect. Then we said variation, gametes, and also maintaining the chromosome number from generation to generation. That's the meaning that the halving effect of meiosis will solve the problem of doubling effect of during fertilization. Yes. So importance of meiosis you have to know. In which structure does meiosis take place in the plant cell and animal cell? We say that here in the anthers, and then also in the ovaries. And in animal cell, we have already answered it. We say that in the testes and then the ovaries. Anthers produce pollen grains. Ovaries produce ovules in plant. Testes produce sperms. Ovaries produce ova. Yes. Then we go the next question. Describe the structure of the karyotype. Here you have to describe what you see. For example, here we see this karyotype. What are you going to describe when it comes to this karyotype? First of all, human karyotype, if you look at it, human karyotype is made up of 46 chromosomes arranged in pairs, each is different from each other by shape, size, and genetic composition. From pair 1 to 12 is called orosomes. The 23rd pair is called gonosome. The gonosomes consist of X and Y, whereby X and X becomes a female, X and Y becomes a male. X is longer than female. So basically you're describing what is inside, which type of nucleus does it have. Yes. Then describe the process of crossing over. Here in the process of crossing over, you describe that homologous chromosomes, they align together. Homologous chromosomes, they align together. One chromatid of adjacent chromosome overlap. One chromatid of adjacent chromosome, or one chromatid of each adjacent, add the word adjacent here, adjacent chromosome overlap with the chromatid of the homologous pair. You understand? It must be adjacent, this and this. This cannot cross with this. It must be adjacent from the homologous chromosomes. Overlap with another homologous pair. This process is called crossing over, and it takes place at a point called chiasma. The chromosomes separate in such a way that each has new genetic material from both parents. So the process of crossing over, that is it. You have to be knowing it by heart. Draw the diagram of the cell after metaphase with the originally with 4 chromosomes. They don't usually bring you this question like this. Sometimes we just bring to say that draw this cell at the end of telophase 1. At the end of telophase 1, then it means that you will be able to, the caption, don't forget to label telophase 1. Remember that now you're going to have 2 chromosomes, from 1 chromosome from there, 1 chromosome from there, 1 chromosome from there, 1 2 3 4 5 6, yeah, and then another chromosome from there, like that, you see. And then the shadings must be right, because here crossing over is going to take place. So you show us at the end, make sure that you label cell membrane, nuclear membrane, chromosome, centromere, chromatid. So you have to label. Don't draw, even if they don't ask you to draw, make sure that you draw and label. At least you'll get, if the other things are wrong, at least you'll get the 2 marks for labeling. What is the function of crossing over? It brings about genetic variation which increases the chances. Yes, what's the function? Brings about genetic variation. What is the importance of crossing over? Basically, since we say that it brings about genetic variation, this helps the organisms to survive differently in the environment. Yes, if you are different from me, it means that you will be doing things I can't do, so we're gonna survive differently in the environment, meaning that the environment, different environment will treat us differently. That's the meaning. Other than crossing over, what is the other cause of variation in meiosis? Other than crossing over, what is the other cause of variation? So you can talk about random arrangement of chromosomes during metaphase, random fertilization. They say during meiosis. So random arrangement of chromosomes during metaphase, and also mutation because also occurs during meiosis. It can occur during meiosis, like the chromosome mutation. So those are the 2 points, because they said meiosis. So if it is out of meiosis, then you can talk about random fertilization, random mating. You see? Yes. What is non-disjunction and how does it lead to Down syndrome? What is non-disjunction? This is the failure of the chromosomes to separate during anaphase. It can be anaphase 1 or it can be anaphase 2. Give two difference between meiosis 1 and meiosis 2. We see that here when it comes to this, I've not explained how it leads to Down syndrome. So in this case, you have to tell us that non-disjunction occurs at a process at a metaphase, and then non-metaphase, anaphase. Let me explain to you nicely here so that I don't waste too much time. Okay, there's non-disjunction. Yes. So if you look at non-disjunction here, we see that here you have in metaphase they were fine. You see, they were fine like this, but in anaphase, this is anaphase 1, we see that this is supposed to separate to go to one cell, this is supposed to go to one cell. So what happens? They separated it properly in anaphase 1. In anaphase 2, the chromosome is supposed to split, you see, supposed to split, but now unfortunately this one split properly, it went this side and this one went this side. That's why you see this one here, and that's why you see this one here. But the second one, the whole of it was put one side. That's why you see 2 of them here. You see? So it means that the cell, instead of having 2 chromosomes, because this one is supposed to be here, it will have 2 plus 1. Now instead of having 2 chromosomes, it will have, because this other one went to this side, it will have 1 instead of 2. So these ones where the separation happened properly, it had 2 of them, you see. So if the cell has 23 chromosomes, then it means that now this cell is going to have 23 plus 1. That's why I say n plus 1. So it's going to be 24. And then if this cell is supposed to have 23 chromosomes, then it's going to have 23 minus 1, because the other one went to here. So it's going to be 22. And then this one, it's normal, it's going to be 23, it's going to be 23. So now, if this chromosome fertilizes a normal chromosome, this cell fertilizes normal cell, so you're gonna see that 24 plus 23 is gonna be 24 plus 23, so it's gonna give me 47, but yet it's supposed to be 46 because it's supposed to be 23 plus 23. So this condition, if it happens on chromosome number 21, therefore it becomes trisomy 21. You have 3 chromosomes of 21 in that individual. Yes, 2 from this cell and then 1 from the normal gamete. So 3 of them. So trisomy 21. And that condition, we describe it as Down syndrome. So that's why it's saying that each nucleus should contain 23 chromosomes. After meiosis, if non-disjunction occurs, one nucleus will contain 22, while the other will contain 24. You see? Yes, that's what I've been explaining. When either of these fuse with the normal gamete, automatically, if this one fuses with the normal gamete 23, is going to be 45. If this one fuses with the normal gamete, it's going to be 47 instead of 46. And this, if it happens on chromosome number 21, therefore is going to be called trisomy 21 or Down syndrome. Yes, so this is very common in people who are aging up. Then the difference between meiosis 1 and meiosis 2. Meiosis 1, you look at phase by phase. Chromosome arrange themselves at the equator in homologous pair, that is metaphase. Chromosome line along the equator individually, that's metaphase 2. You see, the whole chromosome moved opposite pole, that is anaphase. The daughter chromosomes or chromatids moved opposite pole, that is anaphase 2. You see, 2 cells are formed at the end of meiosis, that is telophase 2. 4 cells are formed, telophase 1. Then 4 cells are formed, so that is telophase 2. You see, chromosome number is halved, that is 1 nuclear division. Chromosome number is maintained. Here the nucleus does not divide the number of chromosomes, because it is the chromosomes themselves to separate into chromatids. Here crossing over takes place, that is prophase 1, and here there is no crossing over, that is prophase 2. So look at what happens in prophase, what happens in metaphase, what happens in anaphase, and what happens in telophase. Once you know that, then you are able to understand or to get all these answers correct. Then difference between meiosis and mitosis, we say that when you talk about difference between meiosis result in 4 daughter cells. Mitosis result in 2. Meiosis result in the maintaining of the leads to variation. In mitosis there is no variation. In meiosis we see that we have 2 nuclear division. In mitosis you have 1 nuclear division. So you have to know such questions. All right, so we are done with that. Let's go to genetics. Let's go to the questions of genetics. In this plant, it carries edible fruit. Scientists have been studying the inheritance of 2 genes, one for stem texture and another is fruit shape. So because of 2 characters being considered, therefore this is a dihybrid cross. The stem can be smooth, capital N, or prickly, small n, while the fruit shape can be round or elongated. Name the type of cross which is being studied in this characteristic. With this characteristic, so basically it is a dihybrid cross. The hybrid cross. State the dominant characteristic for the stem texture. Dominant characteristic for stem texture. So we see that texture, we have smooth and prickly. So in this case is smooth, so the answer is going to be smooth. Then recessive for fruit shape, which one is small letter? The one which has small letter is the one which is elongated, is the one which is recessive. Then the genotype of plants with prickly stem and elongated. Prickly, remember that prickly is small n, it can only be homozygous recessive, and then elongated with small r. So since now it is a genotype, so I have to combine them together, NNRR, like that. ORRNN. The phenotype of the plant with both genotype NNRR. Genotype NNRR. What is the phenotype? Because of capital N means smooth, and capital R means round. So it's gonna be smooth and round fruit. smooth stems, smooth what? You have to be specific. Smooth stems, and then round what? Round shape, round fruit. So yeah, that's it. Then we go to the pedigree. How many generation? 1, 2, 3. This means that there is interbreeding. This means these are kids. Yes. Okay, since now we know that, let's find out. Okay. The question is saying that, the first question is saying that, how many generations are represented in diagram? So we saw that we are 3 generation, 2, 3. They're saying that, give the genotype of 9. So the one who is a female with the cysts fibrosis. So but they say that they want the genotype. In terms of genotype, is this dominant or recessive? They said it's caused by a recessive allele. Therefore it's gonna be small b, small b. Yes, small b, small b. So this one's gonna be small b, small b. Then they're saying that, explain why both individual 5 and 6 must be heterozygous for cyst fibrosis. So if you look at individual what and what, 5 and 6, 5 and 6, yeah, because they produce kids who are homozygous recessive. So if this one is capital B, capital B, capital B, capital B, there is no way how they're gonna produce this. Therefore they have to be, if one is capital B small b, where's the second B gonna come from? So there is no way. So they have to be homozygous, heterozygous. Why? So that one small b, capital B small b, capital B small b, so that if this and this they combine, they can form that individual, or they can form that individual. Yes. So in this case you can say that both parent A and B have they are dominant. They have both dominant and recessive alleles. Both parents A and B, yes, they have dominant and recessive allele. Since they do not have, why do they have both recessive and dominant allele? Because they are normal. Is it clear? But their children 8 and 9, yes, they have cyst fibrosis, which means that these kids, they have homozygous recessive alleles. They received one recessive allele from each parent. So basically you need to explain that both parents 5 and 6, they are heterozygous. You understand? Heterozygous, meaning that they have a dominant and a recessive allele, and their kids 8 and 9, they have cystic fibrosis, which means that they are homozygous recessive. They received, or they inherited one recessive allele. From each parent, meaning that one from this parent and one from that parent. Hence they had to be with that condition. Using the letters on the diagram show the inheritance of cystic fibrosis when heterozygous parents are crossed. Here they are talking about homozygous parents. If it is homozygous parents, it means that you're supposed to cross both of them homozygous, and they have not talked heterozygous. They have not talked about which parents are those. So it means that I can't use P2 F2. So I'm gonna say P1 F1. Then you tell me that phenotype. You don't forget this phenotype. Then the last one is phenotype. Then that male normal male without cystic fibrosis, crossed with female without cystic fibrosis, female normal without cyst fibrosis. Don't want to write all that here, but you must write, for example. Then they say that genotype, we go to the genotype. I say, what's the genotype? Both are heterozygous. They said both are heterozygous. This word is very crucial. They say the parents. Why do you say both? Because parents. Then you go to meiosis. They must split, capital B, small b. They must split, capital B, small b. These are gametes. I always dress them up. Gametes. Dress them up. Then from there, fertilization takes place. You can draw a Punnett square or you can cross. So I always tell people, draw a bullet, draw a bullet, draw a bullet, draw a bullet. So this with this, you join the bullets. Capital B, this with that, capital B, small b. Now this with that, you start with the capital letter, capital B, small b. Then that with that is small b, small b. Then these are offsprings. We said F1 offsprings. Yeah. So what is the question? Using A and B to show the inheritance of cyst fibrosis. So in this case, that is it. I can say that the phenotype, we have 3 people without cyst fibrosis, 3 people without cyst fibrosis, and then 1 person with the cyst fibrosis. That's all. Then the same question, we can look at this question now, and then you see that is the same story. You can do a genetic cross, and then you see that you get similar answers. The question is saying that the diagram below shows the inheritance of TAR, a rare disease which leads to the destruction of a neuron. It is inherited as autosomal disorder controlled by the allele TA. Okay, sharp. Describe what is meant by autosomal disorder. It's a disorder which is controlled by allele located on autosomal chromosome. Remember, we have the 6 chromosome, and then we have the autosomal chromosome. So it's the disorder which is controlled by alleles found on autosomal chromosomes. Autosomal chromosomes is pair 1 to pair number 22. The pair 23 is the gonosome, which is a sex. How many sons do individual 1 and 2 have? Individual 1 and 2 have how many sons? They're saying sons. It's only this, because this one came and he married the daughter. So the answer there is gonna be 1. Using the individual 3, 4, 7, explain why it could be concluded that TAR disease is controlled by a recessive allele. Why should we individual 1, individual 3, 4, and 7? 3, 4, and 7. Why individual 3, 4, and 7 is, why do we conclude that it's controlled by a recessive allele? So basically, when they ask you such question, you look at the babies, the individuals they have produced. Yes, you look at the individuals they have produced. So in this case, we see that normal, but they produce someone who has the disorder. Let me check and see. Yes, normal people, they produce, it means that this one was heterozygous, and also this one was, it's like the other question, but they are just asking it in a different version. And then they produce kids who are heterozygous, but in this case recessive. Not heterozygous, homozygous, recessive. All right, so it means that this one individual inherited 1 recessive allele from 3 and 1 recessive allele from 4. So individual 3 and 4 are both without the disorder. Yes, but they produce a kid number 7 who is having the disorder. So in this case, we see that if they have the disorder, this and this, so as we have seen that individual 3 and 4, they are both without the disorder. So since they don't have a disorder, so it means that these two individuals who don't have the disorder, they produce a kid who has the disorder. It means that this individual number 7 received one recessive allele from each of them. Which means that this condition is caused by a recessive allele. Then we go to the next question. We are saying that individual 1 and 2 can produce children with 3 possible genotypes. Individual 1 and 2, they can produce kids with 3. Okay. Then they're asking you, list all the genotypes that have 25% chance of being produced. List all that have 25% chance. Oh, individual 3 and 4. So if you look at 3, if it is capital T, small t, capital T, small t, this and this is going to be capital T, capital T. This and this is going to be capital T, small t. This and this is going to be capital T, small t. This and this is going to be small t, small t. So this is 25%, 50. Is it 25%? No, it's 4, 25, 25, 25. So since this is, these 2 is 50, because it is the same. So it's gonna be 1 out of 4 times 100. So yeah, it's 25, and then this is gonna be 2 out of 4 times 100, which is gonna be 50, and then 1 out of 4 times 100, which is 25. So it means that these 2 are going to be 25. So the answer here has to be capital T, small t. No. Capital T, and then small t, small t. The other one, which is capital T, small t, is 50%. Is 50%, while this one is 25%. Question number 6, 2.6. They're asking you, in human, hemophilia is caused by a recessive allele, small x. A woman with heterozygous for hemophilia marries a man who is hemophilic. Use the genetic cross to show. If you look at this, they are saying that in human, it's the same story. In human, hemophilia is caused by a recessive allele, a woman who is heterozygous. It means that it's the same as the other one, but now you're using a carrier. So it's going to be P1, F1, for example, phenotype. Phenotype is going to be a woman, heterozygous, normal, from hemophilia, crossed with a man who is hemophilic, meaning that hemophilic male. So genotype is gonna be a woman is like this, but they say normal. So capital H, small h, they said heterozygous. And then a male is gonna be hemophilic. Male is like that, so small h there. Then meiosis has to take place so that you split, and then you're gonna have capital X, capital H, then X, small h. These are gametes. So I'm gonna call these gametes, and then this one is gonna be small h, and then Y. These are gametes. So now from there, I'm gonna have to say P F2, so that now this 1, 2, 3, 4, this with this. So it's gonna be capital H, small h. Then now this with this. It's the same thing. It's gonna be capital H, small h. Then this with this, so it's gonna be S capital H small h, and then this with this, so it's gonna be X small h and then y. So basically that's it, that's the offsprings. And then they say that use the cross to show the percentage chance of couple having a daughter who is homozygous normal for blood clotting. Homozygous, a daughter who is homozygous normal. Homozygous normal, the chance is 0. There is no homozygous normal here. The chance is 0 because this is normal female heterozygous, hemophilic normal heterozygous. There is no way you're going to produce a female who is normal unless this person was normal. Yes, so the chance is 0%. So it's 0 out of 4 times 0 out of 2 times 100, which gives me 0% chance. Then we go to the next question. They're saying that is about insecticide. But before we go through this genetic, is it genetic engineering? Yes, so let's do it. Corn are crops that have genetically modified to be insect resistant. The table below shows the percentage of using. Okay. Basically, this is an investigative question. Describe how genetic engineering of BT was done. What did they do? Basically, what they do is the gene for the insect resistance is isolated from another species and then inserted into the DNA of the corn, which is then where it is expressed. So basically, they are doing insertion. They do insertion, so they just get the gene for resistance, then they insert it in the corn, and then once it's done, then that corn is going to be able to resist the disease. So if you, for example, you want something which produce milk, and then this one is resistant to disease. So it can produce milk but it's not resistant to disease. This one can't produce milk but is resistant. So I'm gonna go here, get the gene for resistance, then I put it in this cow which can produce milk. Then it means that the resultant I produce is gonna be producing milk and then resistant to the disease. That's what it happens with genetic engineering. Describe the relationship of land use of BT corn amount of BT corn and the amount of insecticide used. So we see that as the land use to grow BT corn increase, the amount of insecticide used decrease. You see the land use for BT corn, you see, as it increases, the amount of insecticide used also decreases. That's the meaning. I say just describe. They don't want to give you, we don't want the explanation, we don't want the reasons behind that. No. They're saying that state other two plant characteristics that are genetically engineered to increase the food security. State two other characteristics. So if you go back to our genetic engineering, we see that long life, drought resistance, nutrients, increased size, crop yield, herbicide resistance, all this. If we go to our distinction material book, we will find all this in there. We will find all that in there. Yes, genetic engineering. This is what they are talking about. Yes, you see, this is what they are talking about. Crop yield, this increase the crop size, pest resistance. This is what they are talking about. Then they are saying that draw a bar graph. Here you can draw a bar graph, but you guys Please take note of this. Always students fail when they ask them to draw a bar graph or a line graph or what. The graph to represent the percentage of land use to grow Bitcoin between 1995 and 2010. You see, 1995 to 2010. So they say that Bitcoin, meaning that they want this. So it means that you draw remember, bar graph has spaces in between, it has spaces in between, and then this is gonna be X and this is gonna be Y, dependent. So it means that I'm gonna put it there as 1995, 2000, 2005, 2010, and then here I'm gonna put this percentage land use grow Bitcoin in brackets percentage. You have to put the units and then you put the caption. Then after that you'll be able to mark and get all the marks. Make sure that at least you know how to do a pie chart, a line graph. I don't think they can bring the histogram because they brought it in the previous paper. Describe the process of speciation. So now before we go to speciation, we have to do the catch up, and then we see what is the catch up all about in this regard. All right, so it's time for catch up. So differentiate between recessive, dominant, phenotype, genotype, and complete, dominance, incomplete. So recessive is allele which can only express in homozygous state in absence of a dominant allele. Is the allele which can only express in the phenotype in the absence of a dominant allele. And then dominant allele is the allele which expresses itself in a heterozygous state. If you have something like this, this one becomes dominant allele. So it's the character which is being expressed in a heterozygous state. A phenotype is a physical description of an organism, while genotype is the genetic description of an organism. Complete dominancy, here it means that if you have capital T, small t, that this is completely dominant over this. So if I cross, this is gonna be this, so it's gonna be, this person is gonna be taller. However much you have a recessive one, so it's gonna be complete dominance is the one where the dominant allele is completely dominant over one allele is completely dominant over the other. Then incomplete is neither of the allele is completely dominant over the other and results in the production of an intermediate organism. So what is a locus? Locus is the position of a gene along a chromosome. And then Mendel's law of segregation, this one describes the alleles separating, that basically states that the alleles for each gene separate during gamete formation. The allele for each gene separate during gamete formation. So that's why you do like this, capital T, so small t, and then you separate them. Then this becomes a gamete. That's what we have been doing. They're saying that disorder which results in the failure of the blood to clot referred to as hemophilia. Hemophilia. What is sex-linked character? Is a character whose genes are carried on X chromosome or sex chromosomes. They are carried on top of X chromosome. What is sex-linked gene? Is an example. What is sex-linked gene? Is a gene which are carried on X chromosome. An example of a sex-linked disorder, you can talk about hemophilia, and then you can also talk about color blindness. How many allele control blood group? There are 3. And then what are they? Is allele for A, allele for B, and allele for the small allele. How many genes control blood group? It's 1 gene. What is that? Blood group gene. A man who is blood group A marries a woman who is blood group B. Yes. What is the percentage chance of producing blood group O? Yes, you have to do the genetic cross now here. P P1, F1, but you have a man of blood group A, the percentage chance is gonna be, I think, 25%. How? Let's just do it a short explanation, but you have to do the whole process the same way a genetic cross. So if you have blood group A, let me just show you here from our distinction material book so that I don't take too much time. Blood group is above, blood group is above. Yes, so it shows that, yes, it's here. So if you are blood group A, yes, blood group A, and you're heterozygous, and blood group B, you're heterozygous, you have chances of producing blood group, or if this recessive allele and this recessive allele meet, you can produce blood group O. So basically, you will see that the percentage chance of producing a child of blood group O, you see that if A is like this and B is like that, then automatically you're gonna have this, you're gonna be A, B, you're gonna have this, you're gonna be A, you're gonna have this, is gonna be B, and then you're gonna have this, the small, it's gonna be small I, small I. So basically you have 1 out of 4 times 100, which gives me 25% chance. That's why I say that it looks to be 25% chance. What is the percentage chance of producing a okay, we have seen it. What is the how many alleles are involved in the hybrid? How many alleles? So basically here, if you look at there are 2 characters which are being involved in this case. 2 what? That's why they call it a dihybrid cross. So each character will have 2 alleles. 2 alleles. So it means that there are going to be 4 alleles. So you guys, you have to take note of that. Yes, that in the dihybrid, we have seen it in our previous question. We have seen it in our previous question when we are talking about the first, I think it was the first question. You see that how many alleles? You have this, you have this, you have this, you have that. So there are 4 alleles which are involved in that case. But how many characters are involved? There are 2 characters. That's why you call it dihybrid cross. What is genetic engineering? When you talk about genetic engineering, it's you are engineering. We say that whenever I talk about engineering, you are manipulating. What are you manipulating? Genes. So manipulation of genes of organisms. Yes, this can be insertion, removal, transfer, all this. That's manipulation by the human, so that the organisms can produce a specific characteristics desired by the by the human. Yes, so that's biotechnology is part of genetic engineering. So give to advantage of genetic engineering. So we have seen this one before, and I don't think it's now a big deal for that. So when you talk about genetic engineering, first of all, we say that medication, you can talk about pest control, you can talk about increase the yield, all this increase the shelf life. Then the disadvantages: very expensive, interfering with nature, potential health impact, unsafe, unsure of the effects. All this, you see. Name the type of the what are the disadvantages? We have seen it. What are stem cells? These are undifferentiated cells. These are cells which are undifferentiated. They have the ability to divide and form, you see, undifferentiated cells that have ability to grow and form other or any tissue in the body. Yes, examples is bone marrow, umbilical cord, tissue, adipose tissue, amniotic fluid. Yes, all these, they can be modified to form other body structures. So we call them stem cells. What are the sources of stem cells? What are the sources of stem cells? The sources of stem cells, we have seen them. What are the uses of stem cells? We see that they are very good in research. Very good in what? Research. New cells, we can do them for new cells. It's also good for replacing those cells which are damaged, cancerous cells, or that are stem cells. They help us. So please don't go to the paper without knowing these stem cells. Then they are saying that what is cloning? What is cloning? This is the production of identical organisms. This is a production of identical organisms. How do we do that? We use what we call biotechnology. Then please make sure that you know I explained this, and I don't want to repeat that too much. How do you produce this doll fish, the sheep, doll sheep? So when it comes to this, you have to get a sheep and you have to get another sheep. The sheep where the characteristics you want to produce, you get the udder. Udder is, or any other body cell, it must be diploid. You understand? You remove, because I want the characteristics of this sheep, so I keep the nucleus. And then this one, I have to get the ovum. Why ovum? So that you're able to get the casing. So I remove the nucleus because I don't want the characteristics of this sheep. And then after that, I bring this nucleus, I combine together using the mild electric shock. Then once I combine them together, I form something which looks like a zygote. It's not a zygote, but looks like a zygote because it is 2N, and the outside part is the ovum membrane. So that is it. So after that, we make it to insert it into this sheep, the surrogate, and then this surrogate will have to undergo meiosis, then form different ball of cells, and then after that we get each pieces of this embryo development, and then we insert in different sheep. Each is going to become a sheep with the same genetic material as the original sheep. So that is it, and that's how it's being done. So why is it blood group not conclusive for paternity test? Because there are so many individuals which have the same type of blood group. There are so many individuals who have the same type of bloody group. That's why. So here there are some questions. Those people who have distinction material book, yes, please use them. Then why is it not conclusive? Yes, it's not conclusive because of or because that many people have the same type of bloody group. So it means that you don't know which man is it who did what. Then they are saying that why is the male more affected by color blindness than female? Why are male more affected by color blindness than female? It's because males they have what we call they have they have one X chromosome, and then females have two X chromosome. So they require two recessive alleles for them to be affected, while male they only require one recessive allele for them to be affected. Female have two X chromosome, they require two recessive alleles for them to be affected by color blindness, while male have one X chromosome, they only require one recessive allele for them to be affected. The structure that receives vibration from the malleus and transmits it to stapes. So remember, don't forget that it is Miss. Okay. Miss, which is malleus, incus, and stapes. So they're asking you A, oscus, B, incus, C, tympanic membrane or tympanum, and then D is round window. So we are saying that we bring it, receives vibration from malleus, and then transmits it to stapes. You see that if it is Miss, stapes to stapes, from malleus. So it means that the answer is supposed to be the middle bone, which is incus. So it's incus which is in that regard. The part of the male reproductive system that forms the birth canal during labor. Which part of the reproductive system is basically acting as, basically we say, the second name for it, for the birth canal, is the vagina, or the second name for the vagina is the birth canal. So the answer becomes C. So if they ask you what is the second name for the vagina, then you say that it is the birth canal. The structure in the amniotic egg that removes wastes. How do we call that structure which stores? Sometimes we say stores wastes. Remember, we are looking at the extraembryonic membranes. In this case, the extraembryonic membrane, you need to know the amnion, what is it for, allantois, what is it for, yolk sac, what is it for, and then chorion, what is it for. So you have to know the function. But the one which is responsible for wastes, it is allantois. So those are 2 marks. The diagram below represents the cochlear implant in the human eye. Which part of the brain is connected to the cochlear implant? To the cochlear implant. So they're saying that cerebellum, corpus, callosum, cerebrum, and then medulla oblongata. Medulla oblongata. So in this case, we have to ask ourselves that what is hearing? Hearing is a sensation. So you come back to the part of the brain. You need to know the function of cerebrum, corpus callosum, cerebellum, and then medulla oblongata. So the part of the brain which is responsible for hearing or sensation, because hearing is also another example of a sense. So the part, it has 3 functions. All of them have 3 functions. So the answer is going to be C, which is cerebrum, because cerebrum receives and interprets all the sensation. Number 2 is a center for higher thought processes. Number 3, it controls all voluntary actions. It controls all voluntary actions. So in that case, it's going to be cerebrum. Then we go to the next question, 1.1.5. They're saying that the cornea and the lens are both. The cornea and the lens are both, number 1, opaque, meaning that they don't allow light to go through. Then transparent, they allow light to go through. And then elastic, they can extend themselves. Inelastic, they can't. So if you look at them, both they allow light to go through so that an image is focused onto the retina. So this cornea and this lens are both transparent. So the answer becomes B, that both are transparent. Okay. Which one of the following part secretes alkaline fluid to neutralize the acidity of the vagina? Remember that these parts were joined together. That the prostate gland, the cowpass gland, and the seminal vesicle were joined together. They perform, in these days, we ask them as general. So in this case, they're saying that alkaline fluid is basically supposed to be done by the prostate gland. Prostate gland. So I have prostate gland. I don't have any other gland. We call them accessory glands. So the accessory glands here, they are how many? They are 3. That is seminal vesicle, prostate gland, and cowpass gland. So in this case, I only have the prostate gland, which marks it to be the most correct answer. So B becomes the most correct answer. Question 1.7 and 1.8 are based on the diagram below. So you need to know how to label the structure. Whenever they give you a diagram, make sure that you know how to label. This is the umbilical cord, this is the placenta, this is the amnion, and then this is the chorion. Which label part maintains the body temperature of the fetus? But the temperature of the fetus is being controlled by the amnion. So I have to look for the part which is linked to amnion. So the answer becomes C, which is amnion. So you see, if you don't know the part, then automatically you won't be able to get the answer correct. Then we go which one of the following functions function of the umbilical vein. Now we talked about the umbilical vein. You have the umbilical artery and the umbilical vein. Umbilical artery takes blood away from the baby, and then the umbilical vein brings the blood to the baby. So since the blood is coming from the mother, then automatically it's going to have a lot of oxygen and a lot of what? Nutrients. And then the umbilical artery, since it's coming from the baby, is going to have a lot of carbon dioxide and a lot of what? A lot of wastes. You understand? Yes. So now we come back here. So what are they looking for? We are looking for the umbilical vein. So it is that one which is supposed to have a lot of nutrients. So curry is useful. Yes, curry is useful products. Yes, from the fetus. No, to where now? No. Because of from the fetus becomes wrong. Carries waste product. No, doesn't carry waste product. Carries waste product. No, doesn't carry waste product. Say that carries useful. Yes, from the placenta. Here they said from the fetus to where? Yet the fetus is the one which requires this. So the answer becomes from the placenta to. Okay, you got the next question. So that is it. We got the next question. They're saying that the normal site of fertilization in human female is what? The normal site for fertilization becomes the fallopian tube. The fallopian tube is where the baby develops from. So that is it. Let's go to the next terminologies. That is the next question. They're saying that the part of the male reproductive system that stores sperm cells until they mature. We have been singing this song that this is called epididymis. We call it epididymis. The process where sperms and ova fuse inside the male's body. So how do we call that? So since it is inside, they talked about the word inside, so I will add the word internal. But sperms and ova fusing together, then I'm gonna say it is fertilization. Then they're saying that the behavior where the organisms spend time and energy to protect the young ones. So how do we call that? We do that as parental care. Parental care. A disorder that occurs because of the body's inability to produce insulin. If you fail to produce insulin, then we're gonna call it diabetes. We call it diabetes. Diabetes. We are diabetes mellitus. We call it diabetes mellitus. The site where oogenesis takes place in human body. Where does the ova being produced from? That is the ovaries. The functional connection between two or more neurons or successful neuron. So we said it's called it a synapse. Synapse. They're saying that the ball of cells that form during embryonic development. When they talk about this, they are talking about the ball of cells which forms during embryonic development. They're talking about this. How do we call this? The ball of cell. So it is automatically, we're gonna say it is morula. Morula. The period between 10 and 12 years when the human sex organ develop. How do we call that? We call it puberty. We call it what? Puberty. Yes, it's a puberty, meaning that you are ready, mature sexually, but you are not yet ready for case of the country. You are still a baby. Yes. So we are saying that finger-like projection that develop from our outer membrane of an embryo after implantation. How do you call that layer? The outer layer. This layer we are talking about. How do you call this outer layer? So we say that the outer layer is called chorionic. They're saying finger-like projection. The layer is called the chorion, but the projections, the finger-like projection, the one which project out like this, so that now you form the fetus like that. Okay, you form a ball of cells, hollow ball of cells, but it has finger-like projection, so we call it chorionic vili. Chorionic vili. Yes, the chorionic vili. So that is it for the case of the terminologies, and then we go to both A and B. The structure in the ear that converts vibration into pressure waves. How do you call that? Pressure waves are being converted by the oval window. If you look at the ear, yes, we have the structure of the ear. We will look at it, but the last bone, that is the stapes, it causes the oval window to vibrate, and then the oval window creates the pressure wave in the inner ear. So that's why we say that oval window is the one which has that function. So it becomes B only. Autonomic nervous system. Autonomic nervous system is if you look at it, which one is autonomic nervous system? It is the sympathetic and the parasympathetic nervous system, the one which are responsible for making your body to be in under emergence, and once the situation is done, the parasympathetic is supposed to bring it back to normal. So the answer becomes B only. So the answer here, don't write B, don't write A, don't write that. No. You're supposed to say B only. A only. B only. Both A and B, none. A only. So if when you are writing it differently, B, A and B, A, we will cross it. And then don't be surprised that you come out of the paper and then thinking that, yes, you have killed the paper, yet in actual sense, it is the reverse. All right. Contains photoreceptors. Photoreceptors are supposed to be found on the retina. Why? The photoreceptors are divided into two. That is the rods, and then that's the cones. The cones are responsible for bright light, bright and color light, and then the rods are responsible for black and white, and basically for dim light. Being if you have well developed rods, it means that you are able to see at night. So the answer becomes here again B. So B only. So it's B only, it's B only, it's B only, it's B only. Okay. Then we go to the next question, that is 1.4, and then we see what is being asked on 1.4. So you have to make sure that you know how to label each and everything here. So this is the urethra, this is the epididymis, this is the Cowper's gland, and then this is the testis. But if you come back here and then we see, you need to be able to label each and every part. So if I give you this, what are you supposed to label? You see, make sure that you know all these parts from all these diagrams and their functions. So If we go back here, let's first answer this paper, then we shall go back to that one, and then we do what we call the catch-ups. So we are saying that give the letter and the name of the part. Here is 2 marks. Even if you don't know the name of the part, make sure that at least you are able to give me the letter. Just pick it if you don't know for sure. You never know. Your guess might be correct. So where does meiosis takes place? In this case, we are looking for the part where meiosis takes place in the testis. So the answer is D, and then it's gonna be the testis. Yes. Then they are saying that parts that form both urinary and reproductive. Urinary, it means that it's used for urine, and then reproductive, it means it's used for reproduction. So the answer is going to be C, which is the urethra. Urethra. So you see that if you know the parts, then automatically you'll be able to know whatever question they're asking you about. Make sure that you know the parts and the functions of each for the male and the female reproductive system. Then they are saying that the structure that secretes fluid which improves the sperm movement. Remember, I said that these ones are now combined together, but I have to look for the part which is being given, and it is among the 3 accessory glands. So in this case, it is A, and the accessory gland here is going to be the cowpass gland. Cowpass gland. It's the cowpass gland. All right. Then we go to the next question. So it means that now the 6 marks are gone, or we are able to get it right. Name the hormone that is responsible for the secondary sexual characteristics. Secondary sexual characteristics will show that now you are almost a man, though you are not yet a man in the country. So it's being controlled by the testosterone. So that is the hormone which is responsible for that. Please don't ask these too much questions that does the spelling matters. Yes, definitely the spelling matters. The spelling really matters. If you don't, then automatically you'll be losing the mark. So we shall go back to our question, the other question, but now let's continue and find out what are they saying here. They're saying that name the parts. Name the parts, letter, and the name of the part. Make sure that you know how to label the parts of the brain. So we are saying that what is A, that is cerebrum. What is B, that's corpus callosum. What is C, that's the pituitary gland. And then what is D, that is medulla oblongata. And then what is D, that is cerebellum. Make sure that you know the functions of each. For example, this center for high thought processes, control voluntary actions, and also is responsible for receives and interprets all the sensation. Controls, it connects left and right hemisphere together. Then we talked about pituitary gland. This one has a lot of functions. It's one of the glands which produce a lot of hormones like prolactin, FSH, LH, TSH. All these are hormones which are being released by the pituitary gland, or what you call the hypophysis. The medulla oblongata is responsible for involuntary actions. And then this one is very important for muscle tone, balance, and equilibrium. Yeah, so at least make sure that you know some of the functions, so that if they ask you a question, you'll be able to answer those questions. Then they're saying that name the letter and the name of the part that is responsible for number 1. Part A, connecting 2 hemispheres together. You see, now that's where, since I know that this is the one which controls the left and the right hemisphere, then I'm able to know the answer. You see, so it means that the answer there is gonna be B. Then they are saying that heartbeat. Then you ask yourself, heartbeat is what? Do you have control over the heartbeat? No. Though some people they say that their boyfriends, their men, they is being controlled by their men, their boyfriends, their girlfriends. No, man, it's being controlled by medulla oblongata, because that is an involuntary action. So the answer is D, and then you tell me that it is medulla oblongata. So make sure that you know how to write all these spellings. Yes, memorizing. Some people they say medulla. Medulla is wrong because they have different parts which are called medulla. So memorizing the cell phone number, my name, your name, or it means that that is a higher thought process. So because in the higher thought process includes the memory, reasoning, and judgment. So in this case, it's gonna be A, and then the answer is gonna be A, which is the cerebrum. You see? Name two parts of the components of central nervous system shown in the diagram above. We only have two components of the central nervous system. We don't have anything else. So we have this part, and then you have the whole part, which is the brain and the spinal cord. Yes, the brain and the spinal cord. We don't have anything else. Those are the two parts which are involved in the central nervous system. They can even ask you, the question is here, how is the brain protected? How is the brain protected? So in this case, we're going to tell us that the brain is protected through meninges, cerebrospinal fluid, and then meninges, cerebrospinal fluid. Then you talk about meninges, cerebrospinal fluid, and the cranium. Yes, and the cranium. I don't want to go to the distinction material book because it will be just taking a lot of time for us. So I rather just tell you that, guys, just go to the distinction material book if you want to know a lot of this meninges. So you see, it's the meninges, the cranium, and then the cerebrospinal fluid. All right, then we continue to the next question, and then we say that this one is done. So you see that this paper was a little bit easy. Yes, a little bit easy. So the same thing that you're gonna write is gonna be a little bit easy. So you're gonna be able to pass the paper. So just make sure that you go through this paper, you add some questions, and also you go to the catch-ups. The catch-ups are here. I will give you the catch-ups so that you're able, once you know the catch-ups, and then you know this at least, yes, I guarantee you that by the time you come out of the paper, you will be the happiest person on this world. But if you don't do what I tell you, then that's please take note of this question. This question is not going to come the way it is here. No, it won't come like this. But there will be an extract. There will be an extract. So the function of this is to show you how do you scoop the information from the extract. You understand? Yes. So read the extract, and then so that you're able to get this information. They're saying that the male, the male toad makes a clicking call in the water to attract the female. The male then grasps the female from the above. The skin of her back begins to thicken. Right. They're saying that the female releases one ovum at a time, and the female can have more than 100 eggs embedded in the skin of her back. The male fertilizes the eggs on her back. The skin continues to thicken around each egg so that the eggs are embedded within her skin. They are saying that the development stage from the tadpole to juvenile takes place inside her skin, and eventually the tadpoles tadlets emerge from her back by squeezing out through the opening in the skin. All right. They're saying, name the type of fertilization that occurs in this regard. So since these things are happening outside the female's body, outside therefore is going to become the external fertilization. external fertilization. Explain two ways in which the tadpole increase the chances of fertilization. In this case, they're asking you how does it increase the chances of fertilization. If you look at it, we shall see that the tadpole increases the chance of fertilization by, yes, if you look at it here, the female releases ovum at a time, and the female can have more than 100 eggs, meaning that by having 100 eggs released, yes, because if it doesn't release a lot, it can be washed away or eaten by the predators. Yes. Then they're saying that the name 2, they said name 2. The number 2, eggs are embedded in the thick in the back of the female to protect the eggs from being played upon by the predators. So I'm not saying that this question is coming exactly, but I'm saying that there will be an extract which is helping you to. So you have to read. Most cases, the first question and the second question, they come from the extract. But in most cases, the last question not usually come from the extract. But if so, then that you are lucky. Name the type of reproductive strategy you see in the embryonic development that can be seen. So in this case, if you look at it here, you just see, you read to understand. You see, you understand, and then after that, after you understand, then you have to apply. So in this case, we see that the eggs are being laid out, and then fertilization takes place, and the development is outside. So if the development is outside the body, and the eggs are just laid, then automatically it's gonna be ovipary. It's gonna be ovipary. You see, if you look at this word in the extract, you won't find it there, but because of the characteristics you have heard from the question, then you are able to know that that question is that. Then we go to the next question, which is asking you, oh, where is the question? All right, yeah, the question is here. It's about reproduction. But before I go to the reproduction part of it, let's talk about this, because we are about to, okay. Once we are done with that reproduction, I'm gonna go to the catch-ups, and then I finish the catch-ups, and then I continue with the questions. So the diagram 1 below represents development of the follicle, and diagram 2 represents the level of hormones during the menstruation or menstrual cycle. Yes, so they're saying that if you look at this, we see this is a corpus luteum, and then this is the ovum, which is being released by a process called ovulation. Then if you look at this is suspected to be estrogen, and then this is suspected to be progesterone, because it increases after day 14, while this one increases after day 7 and then drops towards day 14. So because of that, so I have to say I suspect. They're saying that identify X. What is X? We say that X is an ovum, and then process Y, we said process Y, it is ovulation. So it is ovulation. And then we say this is ovum. There is a hormone responsible for formation of Z. So if you look at Z, it is corpus luteum. That is, it means that the hormone which is responsible for conversion of empty Graafian follicle to corpus luteum is LH. Name 2 parts of the menstrual cycle. Name 2 parts of the menstrual cycle. Meaning, other words, how is the menstrual cycle divided? It's divided into 2. That is the ovarian cycle, and then you have what you call the uterine cycle. So there are 2 cycles: ovarian cycle, which occurs in the ovary, and the uterine cycle, which occurs in the uterus. Give 2 visible reasons why the female is not pregnant. Give 2 visible reasons why the person here is not pregnant. First of all, if you look at the size of corpus luteum is degenerating, it is disintegrating. It was big, it becomes smaller and then smallest. So it shows that there is degeneration of corpus luteum. And then if you look at this hormone B, which I classified it as progesterone, the level is dropping. Why? Because it is after day 14 when that's the only hormone which increases rapidly after day 14, meaning that after ovulation. And then our corpus luteum is being formed. The moment corpus luteum is formed, now progesterone is being produced. Now, since now the corpus luteum is degenerating, automatically the hormone B, which is progesterone, also drops. So it shows that this person is not pregnant because of the disintegration of corpus luteum and also dropping of the hormone B, which we call progesterone. Then we got the next question. Describe the process of oogenesis. This is a process everyone has to be knowing. It must be like a story. Then you tell us. Under the influence of FSH, the diploid cell in the ovary will undergo mitosis to form numerous follicles. Yes, so they form what? Numerous follicles. So only one inside the follicle will enlarge to undergo meiosis. Will undergo meiosis and to form 4 haploid cells. Only one will survive to form a mature ovum. Yes, basically you have to explain that the way I explained it in the our distinction material book. I don't need to repeat this, but I know, guys, that you have to understand that under the influence of FSH, the diploid cell in the ovary will undergo mitosis to form numerous follicles. Only one inside the follicle will enlarge to undergo meiosis, and then of which of the 4 cells, one only survives to produce a mature haploid ovum. So that is it. Don't need to explain to us the primary oocyte, the secondary oocyte. We don't want those. We just want that. Okay. Then we go to the next question. They're saying name 4 functions of placenta. Name 4 functions of placenta. When you talk about the placenta, you only explain what is being asked. In this regard, what are the processes do you have on your body? It will help you to understand all that concept. So for example, I eat, that is nutrition. So placenta is responsible for nutrition of the fetus. Then I excrete, the placenta is responsible for excretion. Then I breathe in and out, gaseous exchange, the placenta is responsible for gaseous exchange. The skin helps me to prevent substance from entering my blood. So basically is the placenta is acting as the microfilter. The body have the endocrine system which produce hormones. Also the placenta is responsible for production of hormones like progesterone after week 12. Yes, so you see that all the processes basically you have is what is being done by the placenta. So it's also is point of attachment between the mother and the baby. So placenta has a lot of functions. So if such question come, you have no reason why you should fail that question. So before I go that, let me just do a catch up with the topic of reproduction so that I know that now I'm done with the reproduction. I'm not coming back to reproduction. But now I'm going to be a little bit faster when it comes to these questions of the reproduction. What is fertilization? is when the haploid nucleus of the sperm fuses with the haploid nucleus of the ovum to form a diploid zygote. So you guys, I'm not going to keep on writing this. You have to go to distinction material book to find out those answers. If you don't, everything is there in the distinction material book. Yes, if you don't find it, please make sure that you can get it from our YouTube channel by clicking join button just below your video you are watching, and then yes, become a member and then be able to access these books. All right. So we go to the next question. Catch up is done. Distinguish between internal and external. Internal, the sperms will fertilize the ovum inside the female's body, while this one, the sperms will fertilize the ovum inside the outside the female's body or the female reproductive system. Identify the characteristics which show external, characteristics that show external. When you talk about the characteristics which show that this individual is external, we see that the organisms you see that the sperms and the ovum are meeting together outside the female's body. If you look at an example of a frog of the question we have just done now, you will see that the sperm and the ovum are meeting outside the what? Outside the female's body. So it does shows that is the external fertilization. So well, internal, so that is internal. It shows that the male, the cock and the hen, the sperms are being inserted inside the what? The hen. Therefore, there is no way how you're gonna see sperms fertilizing the what? The ovum. So meaning that the eggs you eat are already fertilized with the cock's what? Yes, they were external. You can see them that the sperms are fusing with the ovum outside the female's body. All right, we continue without wasting time. What are advantages of internal? The high chances of the sperms to meet the ovum so that there is successful fertilization. Then when we talk about oviparous is when the development is outside the mother's body. And then internal is when the viviparous is when the development is inside the mother's body and the baby is obtaining nutrients from the what? From the mother. So you see that these questions, you have to know them by heart. They are saying that what are advantages of viviparous over and viviparous over oviparous. Here, because the development is inside and then here the development is outside, here there are high chances of the baby to develop to maturity or to be given birth to compared to this, because now the eggs here are exposed. It's easy for the predators to feed on them. So the eggs are being preyed upon by the predators. While here there is some bit of protection to get those eggs, to get that developing baby, you have to first kill the mother so that now you are able to eat those eggs. Unfortunately for oviparous, it is not the case. The eggs are exposed, and number two, they also experience these harsh conditions of the what? Of the environment. Then we have the next question. They are saying that what is the function of amnion? Amnion is basically, it brings about some bit of protection, and if you go to the human body, it has a lot of functions, whereby talk about cushion, you talk about temperature regulation, you talk about free movement, you talk about free movement. So when it comes to the human, it has more functions than in the amniotic egg. Then chorion is very important for gaseous exchange. It's very good for gaseous exchange. It brings about respiration. And then the yolk sac provides nutrients to the developing embryo. And then allantois is responsible for waste products. Then precocial versus atricial. The major difference between precocial eyes are open, it's born prepared, meaning that is cautious about the environment. So it's born prepared, the eyes are open, able to see, walk, defend themselves. Yeah, as if you check there, you'll be able to see what I'm talking about. Yes, you see what I'm talking about. So all this we are done, you see. Now eyes are open, it can walk, it can defend itself, it has feathers, everything. While when you talk about atricial, eyes are closed, it can't walk, it can't defend itself, it can't feed itself, it can't do basically anything. So you look at, you don't just memorize and then give the answers. You look at the diagram they have given you, and then after that you answer according to the diagram they have given you. Don't just answer and then rush and then continue. Identify the characteristic organisms which undergo precocial and atricial. Obviously, if you talk about precocial, we can see in ducks, we will find them, and then atricial, we can see them in humans. So in this case, eyes are closed, eyes are open, down feathers, can walk, it can feed itself, while this one, eyes are closed, can't walk, can't defend itself. So those are the characteristics you are talking about. What is the function of parental care? Basically, parental care is responsible for bringing about protection. That is the major function. And then also teaching the young ones how the nature, like how to behave in this environment. Then we got the next question is why are, you need to know how to label all these parts. Why are testes hung outside the male's body? Basically, we say that the testes hang outside the male's body so that sperms are produced at a temperature which is below the body temperature, which is 2 degrees less than the body temperature, So what does that mean? It means that if it is less than the body temperature, then it means that yes, it means that sperms, when it is too cold, they have to get a little bit higher temperature. When it is too hot, they are released. So they hang outside to provide the optimum temperature for sperm production. Because sperms are being produced at a temperature which is 2 degrees less than the body temperature. This is what it's saying. This is best temperature for sperm production. So they're saying that yes, sperm, you see that you're saying that they hang outside the male body that 2 degrees below 37, meaning that 2 degrees below the body temperature. Then we got the next question, which they are saying that which hormone is responsible for secondary sexual characteristics? The hormone which is responsible for secondary sexual characteristics is what we call testosterone. We saw that. Now from there you need to know each and every part of this. We say this is fallopian tube, this is endometrium, this is ovary, this is cervix, this is vagina, and the second name for the vagina we say that is the birth canal. Yes. Then we go here. If you look at this here, we see that this is vas deferens, then this is the bladder, then we say that this is the seminal vesicles, this is prostate gland, this is the penis, this is the testis, and then this is the scrotum. You see, so now you are able to answer each and every part. Structure of a sperm, you need to know it is the acrosome, which contains, you have to know each and every part and the function. Yes, this is the nucleus, this is the middle piece, and then this is the tail. When we come back here, we shall see that A is the placenta. It's a placenta. It's the part of the mother's blood vessel, and then here it shows that it's the part of the baby's blood vessels. Okay, and then this is the the uterine wall, and then we see its amnion. This is the fetus, and then this is it looks to be the it used to be the chorion, and then this looks to be the endometrium. Yes, and the endometrial wall, and then this is the what we call the umbilical cord. Yes. So, yeah, you need to know that, and also need to know what happens during the fertilization process here. Then when we come to this, we see that identify the cycle above. This is the ovarian cycle. Remember, we said that there are two. You have the ovarian cycle and the uterine cycle. Which process occurs at B? We see that this is ovulation. And then what is structure D? We say the structure D is immature ovum. And then they are saying that which hormone is produced by structure A? Structure A is Graafian follicle, therefore is going to be estrogen. What is A? That is mature Graafian follicle. What is woman? Which woman is produced by C? This is supposed to be which hormone, not woman. Which hormone is produced by C? It is progesterone. And structure C is corpus luteum. It is corpus luteum, so the hormone is progesterone. Which hormone is responsible for development of A? What makes this to develop is FSH. So here we see that this is FSH. Which hormone is responsible for development of C? It means that after this has been released, which hormone is responsible for this? And is LH. Is the individual pregnant? No, the individual is not pregnant. Why? Because we see that there is degeneration, there is integration of corpus luteum. Give a reason for your answer. There is degeneration of corpus luteum, or the corpus luteum is integrating. Yeah, did I say integrating? Disintegrating. Yes, so it's basically developing down, it's breaking down. All right. Then they're saying which hormone is responsible for stimulation of maturation of sperms? We say that that is testosterone. What is the general name for seminal vesicle, prostate gland, and the corpus gland? We say this is called accessory glands. We call them what? Accessory glands, you see? Yes. Then they're saying that which cells are responsible for production of testosterone? We call them cells of Leydig. Cells of what? Cells of Leydig. They are here. Testosterones are being produced by cells of Leydig. So that is the spelling. So don't say that you don't know the spelling. A little bit faster so that I can go back to our question paper, because this is a catch-up. They're saying that which cells are responsible for providing nutrients to the developing sperms? We said Sertoli cells. Sertoli cells. So if you come back here, we see that the cells are here, are responsible for nourishing the developing sperms. Then we go to the next question. They're saying that will the person's when the person's vas deferens is cut, will the person be able to impregnate a woman? The answer is no. The answer is no. Why? Because the sperms cannot. They're asking you that if this part is cut, will the person be able to? Let's go back to this question. Where did I say? They're saying that if this part is cut, will the person be able to impregnate? The answer is no. Why? Because the sperms cannot move to ejaculatory duct. You see? Why? Because it is cut. Yes. Then they are saying that will the person be able to spread HIV? Yes. Give a reason for your answer. Because there are some other fluids which are being produced. Therefore, these fluids, they might contain HIV. Yes. So other fluids, including blood. Remember, during that action time, there is friction which is being generated. There is exchange of blood. Therefore, that blood can make the person to exchange the what? The HIV and other fluids from the accessory glands. They are saying that if fallopian tube is cut, will the person become pregnant? The answer is yes, because here they say the fallopian tube, not fallopian tubes. So the answer is yes, because the second fallopian tube can be used to produce an ovum. Yes. If one fallopian tube produces one this time, the next one is going to produce the next time. Yes. Give two male and female secondary. Give two male and secondary sexual characteristics. Males talk about the voice, talk about the beard, talk about the the beard, the armpits, the hair in the armpits, pubic hair. The same story to the females, the same story, but you can onset of the menstruation, talk about the development of the mammary glands, that is the breast. Don't call them boobs. I know you guys like to call them boobs, not in life science. They're saying that which hormone is responsible for male secondary sexual characteristic? It is testosterone, and then female, it is estrogen. Estrogen. Describe the process of spermatogenesis. Yes, this one we talked about the process of oogenesis, but spermatogenesis is also the same. Under the influence of testosterone, the diploid cell in the seminiferous tube will undergo meiosis to form four haploid cells, which mature to form sperms. Under the influence of testosterone, the diploid cells in the seminiferous tube will undergo meiosis to form four haploid sperm cells. That's it. Nothing else. Nothing else. Please do that. Please make sure that you have enough time to go through this. Make sure that you go through these questions. Prepare yourself. Don't wait for the last minute, and then make sure that you go through them and then you get the answer. What is spermatogenesis? Is the production of sperms. What is oogenesis? Is the production of ovum in the cells or in the male or female reproductive system. A combination of sperms and spermatogenesis and oogenesis is called, because you are forming gametes, so they're going to become gametogenesis. Yes, that's the combination of the two. That is what you call gametogenesis. So you guys use this distinction material book. I say that get this book from the YouTube channel by clicking join button. Then you will be able to access that book. Once you unlock it, then you are able to get and access these books for grade 10, 11, and 12. All right, then we continue to the next question. Draw the structure of a sperm. Make sure that you know how to draw the structure of a sperm. Why should I draw the structure of a sperm when I saw here that I have a structure of a sperm? So make sure that you know how to draw the structure of a sperm like this. Make sure that the head is proportional to the middle piece and is proportional to the tail. Don't just draw and then leave. Yes, and then make sure that you label. Then they are saying that how is the sperm adapted to its function? What is the function? Talk about the tail having the tail having the is long to provide the propulsion force. Talk about the middle piece providing the having mitochondria to provide the energy for movement of the sperm, and then talk about the head, which has the acrosome, which contains the enzymes which digest the membrane of the ovum. Please take note of this. It is not acrosome which digests the membrane of the ovum. It is the enzymes which are found in the acrosome. So it contains the enzymes which digest the membrane of the ovum. And then talk about the nucleus, which contains the nucleus. So you have to talk about each and every part which you see here. Yes, the nucleus, it contains what? It contains the genetic material for inheritance. Yes, so you have to talk about each and every part. Then we go the next question. They are telling you that how is this? Okay, we are done with that. How is the sperm adapted for locomotion? Now, when you talk about for locomotion, we only talk about here the middle, this tail long enough to provide a propulsion force. Talk about the middle piece having the mitochondria for energy also. Talk about the pointed shape to reduce friction. Yes, and then if they ask you how is adapted it for fertilization, here you only talk about the head, which is it has the acrosome which digests the membrane of acrosome, which contains enzyme to digest the membrane of the ovum, and then the nucleus, which contains the genetic material for inheritance. Then we go to the next question. They're saying that give the function of each part. Give the function of each part, meaning that you are able to label each and every part here. If you look at it, we see that what is A, what is happening here, it is fertilization. What is B, it is the nucleus. As the nucleus, and then what is B, it is a zygote, which process this is mitosis, and then what is this is the morula, and then what is this is the blastula. Yeah, the outer membrane is called the chorion, and then it's called the chorion, and then this is basically the fetus or developing. All right, they're saying that give the okay, what is the consequence when the sperm has no acrosome? Then if it has no acrosome, it won't be able to penetrate the membrane of the ovum so that fertilization can take place. If the acrosome is not there, then definitely, because they can even bring a diagram whereby the acrosome has a problem and ask you what are the consequences. So you have to know that. List the 4 hormones which are involved in the menstrual cycle. So we know this one, LH, estrogen, progesterone, and the FSH. So all these hormones, you must know that this and this, they are from the brain. These two, they are from the ovary. Is the person pregnant? Is the person pregnant? Now, if you look at this small graph, where's my graph? Where did I put my graph? Where did I put my graph? My graph is here. Is the person pregnant? If you look at it, we saw this such question. Yeah, is the person pregnant? If you look at this, the person is not pregnant because the level of progesterone is dropping and corpus luteum is also degenerating. Then they're asking you, so these are questions which are linked to that graph which you're supposed to answer. Then they are saying that give the okay name for okay, is the person pregnant? Okay, from the graph give a reason for your answer. We said the level of progesterone is dropping, and then corpus luteum is also doing what? Is also degenerating. What is the influence of the level of progesterone after 24 days? What is the influence of increase in the level? If there is an increase in the level of progesterone after 26 days, then it means that it will prevent the production of FSH. If increase in the level of progesterone will lead to the decrease in the level of FSH. You understand? Yes. Why? Because progesterone will inhibit the production of FSH, and then automatically the person cannot become what? Pregnant. Yes. What is copulation? Insertion of the penis inside the what? The vagina. What is fertilization? We saw that is when the haploid nucleus of the sperm fuses with the haploid nucleus of the ovum to form a diploid zygote. Explain the process of fertilization. Here they're asking you to explain the process of fertilization. That's when the sperm, the haploid nucleus of the sperm, is when the sperm approaches the ovum. The acrosome of the sperm will release the enzyme which will digest the membrane of the ovum, so that the haplonucleus of the ovum of the sperm can fuse or can enter and fuse with the haplonucleus of the ovum to form the diploid zygote. Yeah, basically that's what they are looking for. If you look at our distinction material book, they are basically asking you to explain this process. After haploid sperm, after ovulation, the haploid sperm produces the enzyme, and then from the acrosome, which digests the membrane of the ovum, and then it penetrates the haploid ovum, and the nuclei, the two nuclei of the ovum and the sperm fuse to form a diploid zygote. So that is the process of fertilization. So if they ask you to label this, you must be able to know how to label. These are sperms moving towards the ovum, you see them, and then if fertilization is taking place. No, fertilization took place. I think you see it has taken place here. It's taking place here. Yeah, so the sperms are moving like this. Fertilization has taken place. It means that now this has become a zygote. So it is moving. This is a ball of cells, which is morula, and then it's still now full morula, and then here fertilization has implantation is taking place here, has taken place here. So a lot of questions you can please do, and then they explain the process. Yeah, we are done with that. So from here it is the the human response to the environment. Okay, let's go to questions which are linked to that, and then we can be a little bit faster and we are able to finish them in just a few seconds. So we have seen the brain, so we have seen this. Yeah, so we are here on our question paper. So the diagram below shows the reflex arc, meaning that the path traveled by the impulse during the reflex action. The arrow indicates the direction of the nerve impulse transmission. If you look at this, we see that this is the hand, therefore this is the sensory neuron, then it enters via the dorsal root. Here is the ganglion, then here we see the synapse. Here we have the interneuron, then you have the motor neuron, then you have the ventral root, here, and then here we have the motor neuron. Yeah, basically, yes, motor neuron, yes, and then this is the effector. So once we know that, then you are able to label part B. We say that part B is the spinal cord. The whole structure is the spinal cord. Define the reflex action is a rapid automatic response to the stimulus. You want to see the word rapid. You want to see the word automatic. Or we can use the word involuntary. Involuntary, yes, rapid automatic response or reaction to the stimulus. Tabulate difference between A and D. A is basically the motor neuron, A is the sensory neuron, and B and D is the motor neuron. So if they ask you such question, make sure that before you think anything else, draw a table, and then this table you write neuron A, and then neuron B. Once you do that, you get 1 mark, and then now you go look for the other 4 marks by looking at the differences between the two. So if you look at neuron A, the cell body, yes, is the cell body is in the middle of the neuron. While neuron B, we see that the cell body is at the end of the neuron. So because if you look at it here, the cell body is here, you see, while here the cell body is at the end. So it means that if you look at the structure, the two structures, let me just use the distinction material book for that structure to be, yep, that is also an example. Or we can go back here and then we see it clearly that they are looking for the difference between neuron A and then neuron D, of which this is neuron A and then this is neuron D. So they're asking you the difference between the sensory neuron and the motor neuron. So talk about the dendrite here, the cell body. Here, check the cell body where it is. You can talk about the dendrite along and the axon is short for the neuron A, which is the sensory neuron. If you look at it here, you see, and then when you look at the motor neuron, we see that the dendrites are short and the axon is longer. You see, dendrites are short while the axon is longer. Yes, so you can talk about monopolar, the other one is bipolar. This is monopolar, it has one side, and then here it's bipolar, it has this side and this side. So any differences between sensory neuron and motor neuron, but please make sure that you know how to draw either the motor neuron or the sensory neuron. We got the next question. They are saying that explain why the brain is not involved in the reflex action. Basically, the brain is slow. The brain is slow. It requires to think before the reaction is brought about, and then by that time, the body is having already the damages. Yeah. So what we do is just do with the reflex arc, the spinal cord, because the spinal cord is a little bit faster. Yes. So it shortens the reaction time. So if the reaction time is longer, then automatically you're gonna have those problems with the protecting the body. Remember, the function of the reflex action is to protect the body from further harm or from danger. If the person suffers from multiple sclerosis, explain the effect of the disease. Basically, what happens if someone is suffering from multiple sclerosis? Remember, this is the disorder which is as a result of the degeneration of the myelin sheath. If the myelin sheath is broken down, then automatically we're going to see that if the myelin sheath is broken down, we're gonna suffer from what we call the slow, because the function of myelin sheath is to speed up the transmission of impulse. So if the myelin sheath is not there, then it means that the speed at which the impulse is being transmitted is going to be what? Is going to be lowered. Hence, we can say that it will cause the transmission of impulse to be slow, and then due to the damage of the myelin sheath, resulting in the slow reaction time or reaction. All right, then we go to investigative question. I'm not going to go through this that much, but you need to read in the investigative question. Make sure that you read the first statement and the second statement. Those two statements are very important. One of them will help you to understand the concept. Okay, the reproductive hormone estrogen and progesterone increase the thickening of the endometrium during menstrual cycle. Okay, an investigation was conducted conducted on the influence of estrogen and progesterone on the thickening. You see, estrogen and progesterone are independent, while the thickening is dependent on the level of estrogen and progesterone which is present. So if the level of progesterone and estrogen is low, automatically the endometrium thickening is going to be low. If it is high, endometrium thickening is also going to be high. So you have to read, and then you see identify the independent. I've just talked about them. So you always get the independent and dependent variable there, or you can get it from the table. Explain the significance of group 1. In most cases, if you see group 1, which group is always the control experiment. Here they are saying that no hormone was given or was given to this group. So automatically this is good for control experiment, to see the other what is the cause of the changes we have seen, because the control has no effect, like it's supposed to run normally. So now any change which happens outside the control, it's being caused by the condition you have exposed to the other experiments or the other groups. State 2 ways in which the validity of the results was ensured. Remember, this is the language was ensured. It means that you have to look at what did they do. What did they do to ensure this? So if you look at it, they're asking you explain the validity. Non-reliability, what did they use the same? If you look at it, we will see that they use the same number of people, same period of time, same individuals, same deity, same you see that what did they use as same. So that is what is going to make you to know that is validity. So they use the same diet, they use the same exercise, they use the same period, they use the same health individuals. You see, yes. State the conclusion of this. State the conclusion of this investigation. So in this case, they're asking you the conclusion. If you look at this, you have to, I'm not saying that the question is going to come like this, but there will be an investigative question whereby they're going to ask you to draw. You understand? So in this case, but I will do a small repeat about it. They're saying that state they want the conclusion. If you look at this, we see that estrogen and progesterone increases the thickening of the endometrium more than estrogen and estrogen and progesterone increase the thickening of what? Of endometrium. Yeah, if you look at this, it shows that If you look at that and you look at that and you look at that, this is the control. So if you check this, check this, check here, check this, check here, check this, check here, check this, check here, check this, check here. You see that estrogen and progesterone increase the thickening of endometrium more than estrogen and progesterone alone. If you combine progesterone and estrogen and progesterone, the thickening is going to be much more higher than when you only have estrogen independently and progesterone independently. That's the meaning. Yes, if you check there. So it's an investigative question. You can have a lot of them. We don't usually repeat such questions. Then let's go to the ear. Make sure that you know how to draw the ear. Remember that the ear, it goes like that, and then you have that part so that now it's complete. Yeah, so here we have the cochlea, you have the autonomic or auditory nerves, which go to, since they are coming from the cochlea, then it means that they are going to cerebrum. And then if they are coming from here, it means that they are going to cerebellum. You understand? So this is the semicircular canals. These are oscus, malleus, incus, and stapes. And then this is, because it is round, so it's a round window, while this is oval window. This is the eustachian tube, and then this is the tympanic membrane or tympanum, auditory canal, and then this is the pinna. So you have to know how to label and give the answers for that. Give two types of receptors in A. If you look at A, we need to find out, is it a cochlea or it is a semicircular canal? So basically it's a semicircular canal, so the answer becomes cristae and maculae. So in this case, I'm going to say cristae, and then I'm going to say also the maculae. Maculae. Yes, so those are the sensory cells. Remember, cristae is speed, and direction, while maculae is for position of the head of the body. Yes, so you need to know this when it comes to the balance, changing the speed and direction of the head stimulates the maculae, stimulates the cristae found in the ampulla of the semicircular canal. Change in the position of the head or the body will stimulate the maculae found in the ampulla, found in the circulus and the triculus of the semicircular canal. The stimuli is converted into a nerve impulse, which is sent along the auditory nerve for interpretation to restore balance. Yes, so basically you need to know balance. Don't make a mistake to go without balance. They're saying that the neuron B transmits impulse to the cerebrum. Okay, neuron B. Then they are saying name, identify, automatically is gonna be, we said that this is auditory nerves. Then they are saying neuron B transmits the identify. Yes, so it's the auditory nerves. Yes, the auditory nerves is a part of sensory neuron. Yes, is a part of what? Sensory neuron. Give one function of D. One function of D. What is D? Oh, it eases the excess pressure from the inner ear. It eases the excess pressure from the inner ear, or you can say it absorbs the excess pressure from the inner ear. Either way, it is correct, and we can mark it. We can mark it correct. Explain the role of C. What is C? Role of C in the interpretation of sound. When you talk about the role of C, remember, role of C is cochlea. So C is cochlea, which contains the organ of corti, stimulated when there is a change in the environment. So basically C is the cochlea, which contains the organ of corti, which is stimulated and converts this stimulus into a nerve impulse. Yes, into a nerve impulse. So basically it's to convert the stimulus, that is the pressure wave, which is generated in the inner ear by the oval window, Yes, the oval window creates the pressure wave in the inner ear, which stimulates the organ of Corti in the cochlea, and then that is converted into a nerve impulse. So that's why we say that cochlea contains the organ of Corti, or C contains the organ of Corti, which is stimulated when there is a change, or when there is a pressure wave which is created in it, and then it converts this pressure wave into a nerve impulse. So yeah, that's it. Give a reason, give one function of D. Give one function of D. So we have seen that. Explain the role. We are done with that. So in that case, we shall say that, yeah, at least. Then question 3, we see we have to label everything. I told you that once you have a diagram, make sure that you label. That is the aqueous humor, that is B, it is the cornea, and then that is C is the what? Iris, and then that is the lens, and then that is the retina. So that is it. They're saying that fluid A. What is fluid A? We have seen it, we have seen it, we have seen it. Explain two structure adaptations of E. If you look at E, E is a lens which is elastic. So it is elastic. Yes. What is it for? Yes. It helps. It's basically the lens which changes the shape. It's elastic. It changes the shape for accommodation, to be able to focus near and far object. Yes. So it is elastic, so that is able to change the shape, its shape, during the focusing of the near and the far object. Number 2, if you look at it like this, it's biconcave. It's biconcave, or did I say biconcave? Because concave is like this. It's biconvex. It's biconvex. That shape is convex one side, and another side is biconvex. So even if you say convex, it's also still fine. What is it for? To focus on light to the retina. To focus light onto the retina. Yes. So basically it does that so that it's able to bend the light to the retina, to bring the light into focus, so that you're able to see the object. Then you can also talk about is transparent, to allow light to penetrate to, so that you are able to form the image to the retina. So it is transparent, to allow light to pass through, so that an image is focused onto the retina. So basically, if you look at the lens, it's elastic, it's transparent, and then also the biconvex shape. All these, we remember when they say explain the adaptation. Adaptation is the part, the shape, and the function of that specific characteristic you have talked about. Don't just say that it's biconvex, it is thick, then you stop there. You just get 1 mark, and then that's all. Describe the role of C. The role of C, that is the iris, in the pyrrhine mechanism when exposed to dim light. Remember, in dim light we have R with the C, and then C with R, of which C, structure C, which is the iris, has sacral muscles and radial muscles. So we shall say that the sacral muscles of the iris relax, the radial muscles of the iris, that where the iris must be there. Radial muscles of the iris will contract, the sacral muscles of the iris will relax, the diameter of the pupil will increase, and then more light will enter the eye. So make sure that you know pupillary mechanism. Then we go to describe the changes that take place in the eye to be able to read the object on time, or on the watch, the watch which is put on the hand. All right. So in this case, the wristwatch is less than 6 meters. So it means that this is the nearby object. So this is dim light, and then this is the accommodation. So in this case, they're asking you to do that. And then in that case, you have to explain the nearby object. In this case, we're gonna say that, remember, when a nearby object, the lens is supposed to be big, meaning that the suspensory ligament must become slacken, and then the ciliary muscles must contract. So we shall say that the ciliary muscles contract, the ciliary muscles will contract, the suspensory ligament will slacken. The tension on the lens decreases. Yes, the lens becomes more convex. The light rays is bent more. The light rays is bent more, and then a clear image is focused onto the retina. Yes, radial muscles, ciliary muscles contract, suspensory ligament slacken, tension on the lens decrease, the lens becomes more convex, the refraction power, meaning that has increased, meaning that now the light rays are bent more to the retina, are bent more, a clear image is focused onto the retina. So that's it. Accommodation, you can't go without knowing periodic mechanism, you can't go without knowing accommodation. So that is done. We go to the, if Andrew looks at an object that is close to him, he sees a clear and with the focus. When he looks at the objects far away, they are out of focus. Identify the visual defect, which is this. Since looks at an object that is close to him, he sees them clearly, but when it is far, then it is meaning that you are able to see object which is near. So therefore it is a nearby object, so which is short sightedness. Yes, this person is suffering from what called short-sightedness. Then they are saying that, explain how Andrew sees the distant object to be cut out of focus. So if you look at it, the eyeball is too round, meaning that now the object will form before the retina. You understand? Our eyeball is too round, resulting in the light rays falling in front of the what? Of the retina. So you won't be able to see, because if it is, the eyeball is too round, in other words, the eyeball is long for them. It's long, meaning that the image, if this is the lens, yes, the image will be focused here instead of being focused here. So it means that the eyeball is too long. Yes, the light cannot fall onto the retina. Yes, so that is it. Then they're asking you, yes, from there we can go back to our catch up, and then we see what is the function of a receptor. Is basically to detect the changes in the external environment. What is the function of effector? We say that to detect, to bring about the response to the environment, the response after you have detected, like the muscles and glands. Yes, those are the effectors. A detectable change in the internal or external environment, how do you call that? That is a stimulus. An electric signal generated by the receptor in a response to the stimulus, we call it an impulse. What is the function of this? The nervous system is basically to coordinate the different parts of the body so that you are able to respond properly, be aware of the environment, protect your body. All that is it. How is the brain protected? Meninges, cerebrospinal fluid, and then cranium. You see, now we're just repeating what we have said. Functions of cerebrum, we say that coordinates all the coordinates all voluntary action, receives and interprets all the sensation, center for all voluntary actions. It receives and interprets of the sensation, controls all voluntary actions, and then is a center for higher thought processes like memory, reasoning, and judgment. Cerebellum, we say that it's muscle tone and balance and equilibrium. Medulla oblongata, involuntary actions, and also coordinates voluntary movements. Corpus callosum, we say that it connects the left and the right hemispheres together, left and right hemisphere together, and also we say that it allows communication between the two hemispheres. It allows communication between the two hemispheres. Then we talked about we talked about how is the spinal cord protected. The spinal cord is protected. We said that the cerebral spinal fluid, the vertebra, and the cerebral spinal fluid, then it is also protected by the vertebra and also the membranes, which are the meninges. Explain what is involved in the peripheral nervous system. Here we are talking about, when you talk about the peripheral nervous system, and then here they're talking about autonomic nervous system. Peripheral nervous system is divided into autonomic nervous system and the somatic nervous system, of which somatic nervous system is responsible for the 12 pairs of the cranial nerves, and then the 31 pairs of the spinal nerves. And then the autonomic nervous system is divided into parasympathetic and the sympathetic nervous system. So when you come back here to our distinction material book, you will see whatever we are talking about here. I just didn't want to remember, yeah, this is what we are talking about. This is what we are talking about, ne? Peripheral nervous system is somatic nervous system, autonomic nervous system. Somatic nervous system involves all the voluntary muscles, and then autonomic nervous system involves the what? Involve the sympathetic and then the parasympathetic nervous system. So we go to continue with our catch up. We're saying that how is the, okay, we are done with that. Nervous system is responsible for storing the body after emergence. We say that is parasympathetic nervous system. Name two systems which work. The two systems work in opposite way. Referred to as is antagonistic. Antagonistic. This is what we are talking about. Antagonistically, two systems work to each other in the opposite way. We call it antagonistic. By the way, that book can be accessed on our YouTube channel. Just go and click on join, and then you'll be able to get the book. Then we continue to the next question. I'm rushing here because these are not. Please make sure that you do all these questions I've given you. Otherwise, you regret, and then I don't want you to regret about it. Yes, the video is a little bit longer, but make sure that yes, you will benefit with it. If you have time for it, you will get the results from it. State 2 system, okay. What is the difference between sympathetic and parasympathetic nervous system? Basically, sympathetic, it brings the body to, prepares the body for emergence. And then parasympathetic, it restores the body from the emergent situation, whereby if the heartbeat was high, so after that, you can draw a table, and then you give us the difference between the two. What does this one do? Heartbeat increase, or decreases, is it? Constriction of the blood vessels of the skin, dilation of the blood vessels of the skin, increase in the blood pressure, decrease in the blood pressure. So that's what we are talking about. Then we are going to draw the structure of a neuron. We did draw it, and then I say that make sure that you know how to draw the structure of a neuron. That's motor neuron and the sensory neuron. What is the function of the axon? Transmits impulse away from the cell body. And what is the function of the dendrite? Transmits impulse towards the cell body. They are saying that what is the function of the sensory neuron? Interneuron and motor neuron. Sensory neuron transmits impulse from the effector, from the sensory cell or receptor to the central nervous system. Connector neuron connects the sensory neuron and the motor neuron, or transmits impulse from the sensory neuron to motor neuron. Motor neuron transmits the impulse from the central nervous system to the effector. What is the reflex arc? Is the rapid is the path traveled by the impulse during the reflex action. What is the reflex action? Is a rapid automatic response to the stimulus. We talked about this. Give two examples of this. Blinking of the eye. Blinking of the eye. You can talk about withdrawal of the hand from the hot object, knee jerk. All that is reflex actions. They're saying what's the function of reflex action? Is to protect the body from danger or from further harm. What is the synapse? Is a junction between two or more neurons. We talked about it. What is the function of the synapse? It protects the body from further harm or from What's the function of the synapse? We say that the function of the synapse is to allow impulse to move in one direction. It allows impulse to move in one direction, and also is a junction between two or more neurons. You can also talk about it. So that's it. the function junction between two or more neurons, sometimes successful neurons. So what is the significance? Ensures the impulse move in one direction, prevents the continuous stimulation of the neuron. You see? Yes. So we continue to the next question. They're saying that what happens if the motor neuron is damaged? So we talked about it. We said that if the motor neuron is damaged, then it means that if your motor neuron is damaged, where is that question? If this is damaged, it means that you won't be able to feel, but you will be able, you will be able to feel, but you won't be able to respond. And then if the sensory neuron is damaged, then it means that you will not be able to feel, but you will be able to respond. How? You can go to our videos where I explained that very well. Then they are saying that describe the process of reflex action. Here I say that you explain the whole process, whereby you talk about this: the impulse through the sensory neuron enters the spinal cord via the dorsal root. You have the synapse, then another synaptic connection moves. The motor neuron moves out of the spinal cord via the ventral root, goes to the effector, and then the effector contracts and then moves out of the. But if you want that, you can also find it here. If you don't have the book, you pause and then you take a screenshot. Yes. You can find it here. This is what I'm talking about until here. So you start here, you go here, you go here, you go here, and then you complete here. You see that the impulse, the body responds to the stimulus quickly, and then from the stimulus, that is the pain. All right, we go the next question. They're saying that write down the path traveled by the impulse during the reflex action. When they say the path, they're asking you that here it is the sensory neuron, the connector neuron, and then the what? The motor neuron. So basically is the receptor, sensory neuron, connector neuron, then motor neuron, and then the effector, showing the arrow going to that direction. I hope you understand what I'm saying, but this is what they're talking about. Yeah, receptor, sensory neuron, connector neuron, motor neuron, and then the effector. That is what you call the path, if they ask you about the path. Differentiate between Alzheimer's disease and multiple sclerosis. This one leads to basically memory loss, the brain cells or cerebrum damaged, and then this one is basically multiple sclerosis, slow in the reaction, and basically is as a result of the degeneration of the myelin sheath. Yes. Draw and label the structure of the eye. We did that, and then we tried to label it, even if you don't know how to draw, but make sure that you know how to label such structures. Yes, and then they are saying that vision involving two eyes, we call it binocular. And then what is accommodation? Ability of the eye to adjust the focal length of the lens so that you are able to focus near and far object. Describe the structure changes which happen to the person of viewing less than 6 meters. The question has just been answered here when we are explaining about the accommodation. This one. Then we are saying that. Just a moment. Then you are saying that what is parallel mechanism? This one is the ability to see in dim and bright light, and this kind of changes to see when the person is in dim light. So the question has been already answered, and they are saying that dim light is C with R. We mix things, C with R, R with C. Radial muscles contract, sacral muscles relax, the diameter of the pupil increase, and then more light enters the eye. Then we talked about short-sightedness. We talked about it. How do you treat it? We treat it by bringing about concave lens. And then this one, long-sightedness, it means that the eyeball is too long. So how do you do that? It means that we do by convex lenses. And then astigmatism, multiple images. How do you treat that? You can do the laser surgery, or we cut with the laser. Cataracts is the crystallization of the lens, and how do you treat that? We do surgery, or we replace it with a synthetic lens, yes, which we call the man-made lens. Then lastly, on this question, describe the process of hearing. When you talk about the hearing, here we describe, we talked about, you explain part by part. The same way here I was explaining to it that you start from the ear, the pinna collects sound wave, directs it to the auditory nerve, auditory canal sends the impulse, sends the wave to the tympanic membrane. The tympanic membrane vibrates and causes the oscars to vibrate too. The oscars will amplify the vibration. The last bone, that is the stapes, will cause the over window to vibrate. The over window will create a pressure wave in the inner ear, and then this pressure wave will cause the organ of Corti in the cochlea to be stimulated to convert the impulse into to convert the stimulus into a nerve impulse. The nerve impulse is going to be moved along the auditory nerve to cerebrum for interpretation to so that you're able to hear. So basically that is it. You have to know that. And then balance, I talked about it. Please make sure that you can use this mode of balance. Changing the spin, the direction of the head stimulates the crystal found in the ampulla of the semicircular canal, changing the position of the head stimulates the maculae found the circulus and the utriculus of the semicircular canal, the stimulus is converted into a nerve impulse. We send along the auditory nerve to cerebellum for interpretation to restore balance. So such questions, you have to be knowing them. There's nothing magic I'm gonna tell you to do. You have to know it. Create a song. Make sure that you remember this. You still have enough time for you to do this so that you master and you kill the paper. Or otherwise, the paper will smash you. All right, if you don't do that. All right, I don't want this paper to smash you. I want you to smash the paper. All right, we continue. What is the middle ear infection? Middle ear infection is basically as a result of the accumulation of the fluid which is caused by the bacteria in the middle ear. Yes. So how do you treat it? We use what we call the grommets. Number 2, we use what we call antibiotics. We use antibiotics. Why do we use antibiotics? It's because this is caused by the bacteria. Yes, we use insert what we call the grommets, and then also add what we call the antibiotics. So the middle ear is being accumulation excess fluid in the middle ear caused by the pathogen infection. That is the bacteria. That is the middle ear infection. Yes. Then we go the next question, which is how do you minimize the hearing loss? Hearing aids, obviously we add hearing aids, remove the cerumen or wax from the auditory canal. Yes. What are hormones? Now we are going to the next point, which is hormones. The hormones. So before we go to the hormones, let's just finish up, and then we finish up our paper, and then we shall go back to the hormones as a what? I call it as a what? As a catch up. So let's go here, and then we shall see. Please make sure that you do this paper so that you are able to benefit in it. So if you try to label everything here, you see here we have the pituitary gland, the thyroid gland, you have the adrenal gland, you have the pancreas, you have the ovary, and then you have the testes. You have to know each and every function of each. That this one produce estrogen, testosterone, this one estrogen, progesterone, this one produce insulin and the glucagon, this one produce aldosterone and adrenaline, this one produce thyroxine, this one produce TSH, thyroid stimulating hormone, LH, FSH, prolactin, 1, 2, 3, 4. There is one which is short, which is TSH, LH, prolactin. Yes. So you have to know what it produces. You have to know, and then growth hormone also produces what you call growth hormone. Yes. So now give the letter name of the part that endocrine gland are responsible for reabsorption of salt. Now you have to know the function of each hormone. Salt it is the aldosterone. So make sure that you know the negative feedback mechanism of salt. Then they're saying that maintaining metabolic rate, that is thyroxine, so it means that it's B. Yes, B. You need to know the negative feedback mechanism of thyroxine. Control the glucose level. Then this is the pancreas, which is D. You need to know the negative feedback mechanism of glucose. So all this, remember, I told you that make sure that when it comes to this negative feedback mechanism, you start with the normal level. Yes, you start with the normal level, level of, for example, glucose in the blood. If there is here increase or high level, the last point must be decrease in the level of that. In this case, it is glucose. In this case is glucose. If here is decrease or low level of glucose, decrease of glucose, then the last point must be there is increase or is rise in the level of glucose. You understand? Then it means that I have two boxes to fill in here and here, and the first box must be about the gland. So in this case, which gland is it? Is the pancreas. And then what is the function of this gland? It produces, since it's too much here, so it's going to produce insulin. It means that gland, and which hormone does it produce? Done. Then now, once you go here, you talk about the function of the hormone you have talked about. So insulin stimulates the conversion of glucose to glycogen. The moment glucose is converted into glycogen, what happens to the level? The level of glucose decreases and then it goes back to normal. So the cycle continues like that. Also 2 points here. If it's decrease here, then automatically I'm gonna say pancreas is the gland. Which hormone does it produce? Glucagon. Now, what's the function of glucagon? Then this one is done. What's the function of glucagon? Glucagon stimulates the conversion of glycogen to glucose. The moment I have glucose, what's going to happen? The level of glucose will increase and then it goes back to normal. So all these negative feedback mechanisms I've mentioned about here, all of them, they work the same way. You just need to know which gland, which normal level increase, the last one is decrease. The first gland, which hormone does it produce? What's the function of the hormone? The moment you know that, done. So basically, you need to know this. What is the gland? Which hormone does it produce? Function of the hormone. The negative feedback mechanism is done. Give the name of the hormone that stimulates the follicle development in the ovary. That is FSH. The saying, give the hormone. Promotes the growth of skeleton and skeletal muscles. That is growth hormone. Don't say this, just write growth hormone. Yes, increase the glucose in the blood. That is glucagon. Why? Because it stimulates the conversion of glucose, of glycogen back to glucose. Name the disorder caused by imbalance of the thyroxine levels in the thyroxine levels. It is what you call the goiter, which I usually tell my students that I call it hoiter. All right, yes, it sounds like Afrikaans, yeah, but it is goiter or goiter, the way how you want to pronounce it, but make sure that the spelling is correct. Explain why the pancreas is considered both endocrine and exocrine. The pancreas is considered to be endocrine and exocrine because it can secrete the hormones and also can secrete the digestive The digestive juices. So the pancreas can secrete the digestive juice and can also secrete the hormones. That's why it's called both endocrine and exocrine. Yes. Then the last question is about the flight. Yes, if not this question, they brought this question last time, so at least make sure that you know carbon dioxide. Yes. They're saying that the diagram below represents a flight or fight or flight reaction considered by the hormone. 3 characteristics of the hormone. First of all, they're looking for the general characteristics of hormone. They are chemical messengers. They are protein in nature. They are required in small amounts. They are transported to the target organs. You understand? They stimulate or inhibit the process. It can stimulate or inhibit the process required in small amounts. They are chemical messengers. They are protein in natures. That's it. Any of that, you can get a mark. Identify the hormone responsible for fight or flight reaction. That is adrenaline. Yes, it's adrenaline. Then they're saying that explain 3 effects of hormone mentioned above. Here is what we talked about even previously, but you can talk about the increase in the heart rate, leading to the more blood saturation or pressure, or more glucose to the muscles, yes, for respiration. Also, you can talk about dilation of the pupil so that more light can enter the eye and you're able to focus properly. You understand? It can increase the breathing and the depth of the breathing so that more oxygen is supplied to the cells for respiration. So you can talk about any other. You understand? Yes. So that is it. Let's go to the catch up and then we see what can we catch up. What are hormones? We said that these are chemical messengers required in small amounts, protein in nature, then transported to the target organs. What is the difference between endocrine gland and exocrine gland? Endocrine glands, they produce hormones. Exocrine gland, they produce other substances like digestive juice and the mucus. Yes. Endocrine glands, they are ductless glands. Exocrine glands, they are duct glands. Give one gland which is both endocrine and exocrine. We said it is the pancreas. They're saying that name all the glands. Make sure that you know how to label all these glands. All these glands must be named. Yes. Right. So we go to the next question. They're saying that the disorder that lack of iodine in the body, that is goiter. We say it is what? Goiter. They're saying that hyperthyroidism is basically it can be as a result of stimulation of the thyroid gland to produce thyroxine, so it becomes hyperthyroidism. The negative feedback mechanism I've talked about for glucose, thyroxine, carbon dioxide, water, and salt. Please use the same principle which I've created here. You will be able to answer such questions. Yes. What are the special cells that are responsible for production of glucagon? We say them is islet of Langerhans. Yes, islets of Langerhans. These are cells which produce insulin and glucagon. You don't need to say beta cells and alpha cells. No, just islets of Langerhans. Then they're saying that what will happen if the person with low level of insulin in the body, basically the person will suffer from what we call diabetes mellitus. And if you have low level of insulin in the body, it means that glucose will not be stimulated will not be converted into glycogen. Yes. Other than glucagon, which other hormone results in the same conversion of glycogen back to glucose? That is adrenaline, because it's also used for adrenaline. It's also used for emergency. Yes, so that more glucose is being produced. What is the hormone responsible for refrigeration? That is the ADH. Antidiuretic hormone. Which structure in the skin are responsible for controlling the temperature? That is the structure, that is the sweat gland and the blood vessels of the skin, which is, so you have to know that. Where are you? Yeah, you have to know that. You have to know. I'm too far now. You have to know the skin, the structure of the skin, and then I'm too far. Yeah, I have to know the structure of the what? Of the skin. I think this thing doesn't want me to show you, but since it doesn't want me to show you, let me just go to the next question. Yes, this. What happens during hot day? What happens during cold day? Then, lastly, which part is responsible for detecting temperature? Describe the thermal regulation on a hot day. Yeah, that's what I said. That hot day, Hypothalamus will send the impulse to the blood vessels of the skin. The blood vessels will dilate. More blood will flow near the skin. More heat is going to be lost. More blood is going to be sent to the sweat gland. The sweat gland becomes more active. More sweat is going to be produced. And then as it evaporates, it will lead to the cooling of the body. Yes. If you look at our distinction material book, this is what they are trying to say. Hypothalamus stimulated sends impulse to the blood vessels. The blood vessels dilate. This is called vasodilation. More blood vessel flow. More blood flows to the skin. More heat is lost from the skin. More blood is sent to the sweat gland. The sweat gland becomes more active. More sweat is released. Evaporation of the sweat cools the skin. Bam. Done. Simple. They're saying that how do I differentiate between the blood vessels of individuals in the cold day and hot day? Blood vessels of individuals in cold day, you see that the shunt here, and the hot day, the shunt here is closed and more blood is flowing near the skin. You see, while blood vessels in the cold day, the blood will go through the shunt and then the blood vessel near the skin will be closed. A very good example is this. You see that this is cold day and then this is hot day. Yeah, that's what it is trying to show. And if they ask you what shows that this is a hot day, you see the sweat and the blood vessels have dilated. They have dilated. When you say validity, look for your answers must include the word the same. Reliability, increase the sample size, but don't just give generic answers. Tell us what did they do. Answer the question depending on the language they have used. Planning stage, make sure that you use the word decide. If you don't have the word decide, you're most likely to have it 0. Decide on the place you're going to do, the sample size, the individuals. Decide, it must have the word decide. Independent, and dependent, in most cases, we get it from the statement, the first statement of the investigative question. So if you, the same way I explained to you in the previous question, and or you go to the table and then trying to check it. In most cases, we can get it from the table, but not always. But 90% we can get it from the table. Control variable, this one is very important. The one which we have not changed, it's kept constant all over. If control experiment, this one is for the function is to compare the results and see what is the effect which is being caused by the other conditions you have altered. Please know how to calculate the percentage increase and the percentage decrease. Percentage increase is final minus initial over initial times 100. That is percentage increase. So you will find out that maybe it's 10 minus the initial, which is 2, divided by 2 times 100. These percentages can be more than 100. It can be like 200 also. So if you get 200, don't be scared that my answer is wrong. Yes, percentage decrease is basically initial minus final over initial. So initial is always down. Yes. Pie chart, make sure that when you go to the pie chart, first calculate. If they give you 2, 4, 10, and 20 as the figures, please make sure that you first add them. 2 plus 4 plus 10 plus 20. This is 6, this is 16, this is 36. So the total is 36. And then now you say that 2 out of 36 times 360, then you get the answer as degrees. You don't plot with this. You don't plot with this. As long as it's not in degrees, we don't plot with them. You go to 4 out of 36 times 360, so that you convert it in degrees, and then 10 out of 36 times 360, you get the answer, and then 20 out of 36 times 360, and then you get the answer. After that, bring your protractor. Make sure that you bring your protractor, and then on the compass, remember it has a point, then it has a proper circle. Yes, if this circle is like this, we'll cross it. Yes, and then this point is where you put a protractor and then start measuring one by one, and then give a key, give a caption. Bars, the bars are supposed to be the same. Please don't make the bars to be different sizes. The different sizes. It's not a must that you supposed always start with a 0. You guys, you always start with 0, and then here you say 200, and then you say 201, 202. No. It's not a must that you're supposed to start with the what? With the 0. Histogram has no spaces, but the bars are supposed to be the same. So you guys, that is it. I think I wish you the best for today. I will stop here. I will come back for paper 2, preparing you for paper 2. Please use this resource. Please, I repeat, use this resource. I know that if you use it, you're gonna get a distinction. As you know that I always tell you that use these resources. Yes.